Hello everyone, and welcome to the second day of our Life Science Summit here at ABG. My name is Gonzalo Artiach, a pharma biotech equity analyst, and I have now the pleasure to introduce the first company of the day, which is OncoInvent, a company developing novel treatments using radiopharmaceutical products for treating cancer. And with us, I have the pleasure to introduce the CFO of the company, Tore Kvam, and the CMO of the company, Kari Miren. Please guys, the stage is yours.
Thank you, Gonzalo. As Gonzalo said, we are a radiopharmaceutical company focusing on the oncology treatment of oncology. And before we go in to talk about the company, I'd just like to start a little bit about how we see the market. I just came back from a conference, radiopharmaceutical conference actually, where most of the companies operating in this space were present. And there is some challenging that we have within the industry that is a highly focused area right now. We believe there is a big opportunity within this space, and it's really at the very beginning of developing radiopharmaceuticals. We see there's more companies coming, more innovations, there's more money getting into those innovations. Currently, most of the targets are very focused on a few areas, but that has to do with the infrastructure that is not in place still. The infrastructure within the industry is lacking for a supply chain in particular. You will see that products that are on the market are struggling to deliver the product to the patient on time when they need it. There has been Novartis have been struggling with that when Pluvicto and so on. So there are some challenges to overcome. Plus, there is also a focus on the isotope. Will there be sufficient commercial supply of isotopes? These are challenging. That is highly focused in the industry now. That we discussed a lot both with suppliers and other similar companies like ourselves. At OncoInvent, we think we have solved a lot of this. There's been a focus for our way of organizing from the very beginning, which I think has given us an advantage. If you look at who we are, we have early on said that, okay, we want to take the space of being an alpha emitter, focusing on that, and that is what the isotopes we want to work with. We have established an organization of 50 people. So we have competency, have been focusing hard on establishing a competent organization that are able to deliver within this space. We're located in Oslo. As I said, 50 people. We have taken full control over the CMC process, where we both produce our own isotopes. We produce our own products for clinical trials. And we do that basically in the city centers. This has created, obviously, a lot of challenges for us in the sense of we have to have control of the radiation at all time and those things, which we do manage. Obviously, as this is the lab that we have established, it's a research and pilot production lab. Once we expand to more advanced clinical trials, we'll also expand the production. We need to increase the capacity, and we need to create a situation of redundancy in production. So that is also a focus of attention for us going forward. Currently, we have 2 trials ongoing. Kari will talk about that a little bit more later. So I won't go into those. If you look at our lead compound, Radspherin, the concept of Radspherin is to treat metastatic cancer in body cavities. That's the whole concept of it. In the simplest form, it consists of 2 components, really. Calcium carbonate microparticles, which we attach radium-224 to. Radium-224 has a half-life of 3.6 days. So obviously, the logistic is important to us. How it gives Radspherin a shelf life of approximately 1 week. So it's produced basically on demand. Both in preclinical testing and later on in clinical testing, we've seen that the distribution of radspherin in the cavity that we're using has been good. That has been a crucial answer that we got from those studies. Because distribution of the radspherin to kind of treat all the cancer in the cavity that we're treating is really the key for the product to succeed. And that, I think I will let the word over to Kari.
Yes, so and as Tore mentioned, we are developing our product to treat cancer in body cavities. And the first cavity that we have started in with radspherin is in the abdominal cavity. And we are treating peritoneal metastasis, meaning all kind of spread of cancers that have spread into the peritoneal cavity. So in the peritoneum, by far most frequent is cancers that come from other organs, and the prognosis is in general quite poor. The treatment options are very limited, and it also represents a very high burden to the patients and a lot of painful symptoms. And as you can see here, also the different, there are some primary cancers in the peritoneum. They are very rare. Most of them, as I said, are secondary to other cancers, and they arise mostly from the intraperitoneal origin. And the three most frequent cancers are colorectal cancer, ovarian cancer, and gastric cancer. And you can see the frequency that peritoneal metastases occur in this disease is varies a bit. And you can see in ovarian cancer, it's as much as 60–70% of all ovarian cancers actually have spread in the peritoneal cavity. So it's definitely an area where there is need for new treatment options. You touched upon it a little bit, but I'll go into some more details. So radspherin delivers alpha radiation, and that means it's very, very highly efficient radiation that breaks both strands of the DNA, means that the cells are not able to repair themselves. As compared, for example, to beta radiation, where you only damage one of the DNA threads so that the cells have a possibility to self-repair. So it's an irreparable damage. And in addition to being very efficient, it only reaches for a very short range, around 100 micrometers. So it means that the risk of damage to adjacent tissue is very, very low. And also it's a point here that the radspherin is used postoperatively, so it's used in conjunction with surgery. So after you removed every visible tumor, there is a problem that's very well recognized, that there are small clusters of cells that arise either from shredding from the primary tumor, or it can also happen that it releases during manipulation during surgery. So it's, as I said, very well recognized then that these micrometastases, they deposit and they can give rise to new recurrent and progression of the cancer. So the aim with radspherin is to, when you do the complete resection surgically, you give radspherin the first or second postoperative day, and then you aim to eliminate all these micrometastases and then to prolong the disease-free period. And it's also a very important point that it's actually, these patients go through multiple rounds of chemotherapy, for example, and they get increasingly chemo-resistant as they go by. So this radiation is independent of resistant and also for dormant cancer cells, and it's target independent. It's also an interesting fact, you mentioned that the half-life is 3.6 days, and together with these slowly degradable calcium carbonate microparticles, this radiation is kept within the abdominal cavity, and it releases continuous alpha radiation over a period of around 1 week. So you get, with a single treatment, you get for a week of treatment. we have 2 clinical studies ongoing in 2 of these major indications that we talked about earlier. One study is looking at ovarian cancer in recurrent patients that had previous treatment. We have 3 clinical sites, one in Oslo, one in Leuven, and one in Madrid, 2 actually in Madrid and Pamplona. And we have another study in colorectal cancer patients. And these patients both undergo surgery and actually treatment with heated chemotherapy after the surgery as well. So quite heavily treated patients, 2 study sites in Oslo and in Uppsala. For both of these studies, we have gone through the dose escalation phases and have selected the highest dose of 7 megabecquerel for both studies. And then we have expansion cohorts ongoing in both of these studies. Now we have treated more than 50 patients in total. And so for the colorectal cancer patient study, we have come a bit farther. So there we actually have this first 23 patients in this first part of the study have now been in the study also for 18 months, giving us some interesting also indications of clinical efficacy. So before we go into that, of course, safety is what we have mainly been focusing on in these first studies. For now, it seems to be very well tolerated. We have not observed any dose-limiting toxicity. We have also done assessment of the doses that is received in all the body organs, and all the dose levels are well below dose levels associated with any toxicity. We have also assessed the clinical administration procedure. So a catheter is left in place at the end of the surgery, and this catheter is being used for administration the first or second day. And everything around the procedure has been very simple and manageable. And we, as I said, we have also done dosimetry in 6 of the patients in the colorectal cancer study to assess the doses to organs. Also, we have looked at the radiation around the patient. Of course, that's an important logistical factor, but also it has been a very good safety profile. And we actually also published the results from the measurements that we have done around the patients. Of course, in addition to safety, we are very eager to look at indications of any efficacy. As we have seen in the animal studies, we have also seen this confirmed now in the clinical studies, or at least get some indications of it. I mentioned already the colorectal cancer study, we have come a bit further, so we now have quite a lot of all the patients that were treated in the first cohorts. They have now been in the study for 18 months. These are the results at 12 months, and will be presenting the 15 and 18 months later this year. Actually, the 15 months data will be presented next week at ASCO. So I cannot talk about it today, but it looks very encouraging. What we know from these colorectal cancer patients that has undergone surgery, they have undergone heated chemotherapy, yet 50% of them will have recurred at 1 year. So median time to progression is around 12 months. What we have seen in the recommended dose of 7 megabecquerel is that only 25% of the patient had recurred at 12 months. Of course, one should be considerate to draw any conclusions. It's a low number of patients, it's quite early on, but still I think it's very promising. And also, of course, we mainly believe that we can achieve local control in the peritoneum, and then we see actually in the highest dose of 7 megabecquerel, none of the patients in the recommended dose had any experiences of peritoneal recurrences. I said already we have published the safety results from around the patients. We have also published earlier this year the first experiences, the first 30 days of from the colorectal cancer study. Last year we presented this at ASCO again on the first initial safety, and this year at ASCO, we'll be presenting the 15 months. Also, the main congress in this kind of specialized area, the SOGI conference in Venice in October, we have submitted the 18 months results. In addition, we have also the ovarian cancer study. We are then only completed with the first kind of 30 days safety that we have also submitted for the main conference in the gynecology oncology space is ESCO in September this year. So, of course, we do find these results very encouraging, and we want to explore them in future studies. For the time being, we have planned to move on with both indications, the ovarian cancer and the colorectal cancer indications. And in ovarian cancer, we actually, as I said, we briefly mentioned that we now are in the recurrent setting, so patients that have already been treated. Our plan is to move it up to the primary population, so meaning that we go in the first time they get operated for the ovarian cancer. For the ovarian cancer study, we have already had the first meetings and discussions with the FDA and got some scientific advice back from the EMA. And I would say more or less they kind of bought into our concept, and we are now finalizing the study protocols with some small adjustments and hope to be able to start a control study. And also for the colorectal cancer studies, we also see that the data seem to be quite encouraging, and we hope that we can request a fast-track program with the FDA and work on finalizing the protocols here as well. Then I think it's back to you, Tore.
Yeah, thank you. If you look at the milestones for the current year, the single most important one for us is to finance the next clinical trials, the one that Aktier just presented. We have been fortunate in terms of financing and been well financed so far. And now we have the results coming up, and we want to use those results to finance the next step again. So the phase 2 programs we've been very clear from the very beginning, those are not included in the finance that we have. As Kari said, we will be presenting next week the 15 months results on ASCO, followed by the 18 months results. In addition to that, we also do have a pipeline of more traditional targeted therapies that we reveal kind of how we will proceed with those. So we haven't been very open on those. Those are currently in a preclinical testing. So that is. And then the last one that I would like to mention is the production capacity. We are currently looking at ways to increase that capacity, and we have always had the idea that we would like to see a production site on the European side and one on the US side to have the redundancy. We're capable of logistically handling the product between the two continents, but prefer not to. We prefer to set it up like that. So with that, I'd like to round off and leave it to the Q&A.
Great. Thank you very much, guys, for such a nice presentation. Now we have a few minutes for questions. And I would like to start with one question around rasferin. And I was wondering how homogeneous is the effect once you inject it or once it's placed in the cavity? And do you have any risk there of developing hotspots of radiation or the other way around, cold spots?
I would say in the studies that we have done, we have also done imaging, gamma camera imaging and SPECT CT imaging to look at the distribution, and it seems to be very well distributed. And there has not been any reports of any cold spots, any untreated areas. And of course, there will also always be some heterogeneity, but also for the hotspots, they are very limited and does not seem to be representing any clinical problems. As I said, we have also in the studies not had any serious adverse event reported as related to rasferin. And as I said, we have also now treated around 50 patients. So, yeah.
Interesting. And the second question also in your lead compound, I was wondering, do you have to radio-label it right before use? I mean, a little bit on the feasibility of the project and what kind of infrastructure do you have to have in the hospital in order to administer it to patients?
Yes, so Ratsferien is produced as a ready-to-use product. So the solution is delivered completely finished from Oncoinvent to the clinical sites. So there's no radio labeling needed to be done at site. So the only thing that they will do is that they will calibrate the dose and dilute it, and it's normally then administered by the nuclear medicine physician or a delegate at the hospital. So it's very easy at the routines at the hospital as well.
Great. And another question. Now you're developing your trials in the metastatic setting. Is there any way you can go to to primary tumors, to treat primary tumors, let's say for colorectal cancer, and combining it with standard of care there?
So I would say the treatment principle or the concept, if you think about it, if you have a large bulky primary tumor, you would not expect Ratsferien to have a treatment effect on that. So we are it's a prerequisite that you remove the bulky disease and you treat the micrometastasis. One concept that you could, of course, think about is something that has been looked at to some extent in colorectal cancer, but more maybe in gastric cancer, is that you have patients where you have a positive cytology, meaning that you have cancer cells detected in the abdominal fluid without having any visible lesions. So that could also be something that you do it in adjacent to surgery, but not as a standalone treatment to bulky disease.
Great. And one more question here. You're running these two trials. I was wondering how big or small is the cross read that a person could do from if you score positive in one trial, how likelihood is that you have also positive result in the second one?
Yeah, so these cancer types are they are a bit different in nature. They have different growth pattern, different prognosis, different treatment. At the same time, if you look specifically at the situation where you have the peritoneal metastasis, the concept is the local control, to control the development of the recurrence and new metastasis in the abdominal cavity. So I think if you look at that concept, you would expect it to happen maybe a bit later or sooner in some of the indications. But still, I believe that if we are able to demonstrate that we do better than the expected standard of care, I think it's definitely something that could be used across indications as well.
Great. And one last question. I would like to take the opportunity to ask you about the future plans for your company. How do you see the company moving forward in the next months or midterm, and any message you would like to bring to investors that they should not miss?
No, as I said, with the milestones, our focus now is to finance the next step. Yeah. So what we're doing now, we're really preparing the organization for entering into a phase 2 study, as we indicated, and then to get the financing for those studies in place. Great. That is where for the short and the short range.
Yeah, great. Thank you very much for being here today with us and for your nice presentation.
Thank you.
Hello and welcome back, everyone. Happy to have Gentian Diagnostics here with us now, and Dr. Hilja Ebert here to present the company for us. I'll let you go ahead with the presentation right away and come back for a short Q&A after.
Thank you very much for the invitation and the introduction. And let's go to the next slide. Next slide, please. Yes, Gentian Diagnostics, as the name says, is a player in the healthcare market, and to be more specific, in the clinical diagnostics market, which is as well called in vitro diagnostics market. Our mission is to innovate the diagnostics efficiency in order to allow clinicians to take better treatment decisions. And the objective of my presentation today is to explain a little bit more in detail what is behind this mission statement and what is the value for our customers and as well for our shareholders. With this, please, the next slide. Let me start with a short introduction to the company. We have about 50 employees, more than 50 employees in the meantime, and we are located in the headquarter Moss, Norway, and we have sales offices in Sweden, in the US, and in China. From those sites, we are serving customers all over the world. Customers, as I mentioned before, are those who are providing diagnostic results to treating physicians. So these end users of our product are laboratories, clinical laboratories. We provide reagents to those laboratories, which can be used on all kinds of instruments which are already established in those laboratories, which makes the introduction of a new product into those laboratories much easier rather than introducing a whole system, instrument, software, and reagents. We served customers and users directly all over the world, but we do that very selectively as we are a small company and we focus on innovation, on technology innovation. But we need to be in touch with key opinion leaders, with universities to know where the needs are. So therefore, we have decided to selectively serve customers directly, but the majority of our revenue is generated via partners, commercial partners, and our preferred partners are those who provide those instrument platforms to the laboratories. So they sell our reagents as part of their total package to laboratories, and that, as hopefully you will agree later with me, works quite well. We have 7 products. 5 of them are in the market. 1 is in market development and 1 in product development, and I will speak as well in a minute about it. We are listed on the Oslo Stock Exchange and having achieved a revenue of 112 million NOK in 2022. Let's go to the next slide. I strongly believe that a company is up for great success if they address a significant problem in the market. The problem which we have identified several years ago in the market is the need for efficiency on the laboratory level, but as well on the clinical level. The challenge for specifically for our customer and the segment we are working is that the laboratories have to serve clinicians with more and more test results as they have more and more patients to serve with the same resources. And in addition to this, the clinicians ask for those results fast, as they would like, while they treat the patient, would like to see the patient, they would like to have results as fast as possible. The many biomarkers, clinical relevant biomarkers, are not available on clinical instrument platforms which allow fast results and high throughput in order to drive productivity for the laboratories. And that is the solution we are offering by shifting relevant biomarkers from inefficient, low throughput and slow platform to the most efficient platforms laboratories have installed in their laboratories. And with this, we are providing them the opportunity to achieve faster results, which again allows clinicians to take those results into account when they decide on the treatment of the patient. So we are especially here in the hospital setting. And we achieve, together with the laboratories, a throughput gain of 3 to 10 times, providing, depending on from which instrument or even manual platform they are changing and shifting the biomarker. So these are, as we believe, high benefits for our customers. And this is as well what we have seen within our performance throughout the last year. So let's go to the next slide, please. And based on this, we have defined a growth plan based on the 7 products which I mentioned before. Those 7 products are addressing a market of about $1 billion in this case, as market research is mainly stated in US dollar. This market is growing by 5 to 10% on an annual basis. Again, I mentioned before, aging population and other reasons. There is that there are more and more patients showing up in hospitals, or as well as general practitioners, and need to be served as well with clinical diagnostic results. So the performance of our test would not be achieved without the team we have assembled here in Moss and as well in our sales offices. We have an industry-leading expertise in the area we are working on, and that is deep technology. And this expertise, unique expertise, which we have in the team, is making as well us very positive for the future. We achieved, as I mentioned before, already commercial partnerships, especially with companies who provide the instrument platforms to the laboratories. And these are normally the top companies in the market, as the development of instrumentation with all the respective software solutions, etc., requires a strong backing. But those companies are as well in heavy competition. And one piece of the competition is to offer a broad menu on the instrument platforms they sell to the laboratories. And here is where we come in, as they cannot develop all the assays themselves. They are partnering with companies like us and broadening their menu via partnerships, which we have established now in the meantime with the top 5 major players in the market. And based on the products, the team, and as well those partnerships and more to come, we have defined a long-term ambition of 1 million NOK for us, with an EBITDA margin of 40%. And I just see that you probably cannot see the footnotes. The footnote means that, of course, this is an ambition, and it is very much dependent on when we can launch those products which we have not in the market, like NT-proBNP, which is still in product development, and as well how fast our other product, which I will speak in a minute about, GCAL, is picking up in the market in terms of acceptance. But having that said, we are positive about our growth plan. When we look back, since we have started to define the plan, since we have started to further strengthen our team and adding products to the portfolio, we have achieved a 28% average sales growth between 2019 and 2022. And therefore, we have believed that our strategy is working, and we are looking positively into the future. Let's go to the next slide. I spoke about the team. Our management team, which I present to you here, is a good representation of the total Gentian team. We have our co-founder, Arling Sundarang, still working for us as consulting founder. And we have many people who started with Arling 20 plus years ago in the company, still working for us. So we have history, we have deep expertise in the technology we are working from, and we have brought additional expertise from companies, the big players in our market, so who had supported those companies to be successful in the past. And we believe that this experience helps in the communication with the big partners, helps in the communication with our end users, and as well to build the required processes internally to serve end users and our partners now and in the future at best level. Next slide, please. I have spoken about our products, 7. And as you can see on the left side, the products are addressing all kinds of disease areas. We have not picked those products based on disease area. We have picked those products based on how can we, by shifting those biomarkers from the current platforms to the clinical chemistry platforms, drive efficiency from a laboratory point of view and as well from a clinical point of view. That is the decision-making matter for us. So all of these products, you can see, provide the productivity gain for the laboratory, and all of them have very specific clinical benefits. Like when we just take the first one, cystatin C, using this biomarker and providing results to clinicians early means that ultimately it can prevent severe kidney failure. And there's a long story, but due to the shortage of time we have today, I cannot go into the clinical benefits of cystatin C and of all the other products. But important is that we have clustered them in 3 different categories. One is the category where we have cystatin C and as well the next one, FCAL, for example. Those products are already very well established in the market. The knowledge is there. We are just supporting, from a commercial point of view, our partners to even gain more attraction and traction for those biomarkers in the market. Established, can be ordered, are reimbursed. We have with GCAL, it's the 3rd product on this list, a calprotectin assay, which detects severe infection and inflammation earlier than any other biomarker. But this biomarker is not yet so fully established. It is scientifically very well described. The value proposition is as well proven. We are confirming as well with additional studies the clinical benefits of such a biomarker. And this goes its way through market development, which will take time. And it's very country specific. We started our own efforts for the market development in Sweden. And we can see now in Sweden, this is picking up, and as well in other countries where we have started with studies some years ago. So this is a product which is a perfect example of what a company can achieve, even a small company, with market development, when you follow the science, when you have the science behind you. And the 3rd category is the anti-proBNP product, which is addressing the cardiac disease group, a very well-established and very powerful biomarker. There are many reasons why we believe that this biomarker would bring a lot of benefits for laboratory, but as well for the clinicians by bringing it to the clinical chemistry analysis. This product is in product development. It is a breakthrough product because we would achieve sensitivity levels which nobody ever had achieved. And therefore, this is a product development which takes time, which takes longer than our average, and which has as well a level of uncertainty, which we have built as well into our plan, which I will describe a little later. But this is as well the uncertainty we have on when do we achieve our billion NOK ambition. Next slide, please. And that is what I mentioned in terms of value creation based on our business plan. We have, if you look to the right side, it's a very busy slide. If you look to the right, the billion NOK ambition with the 3 product categories: established product on the bottom, GCAL, the infection and inflammation marker in the middle, and pipeline product, which is mainly anti-proBNP on top. And if you add up our ambitions for those 3 categories, you get to our billion NOK long term. But there is, of course, level of uncertainty where you can see that with established products as well, when you then look a little bit to the left side, you see that in the years, in the quarters and years before, we have achieved good traction with those products, and therefore we are very positive with the performance we have today. We doubled our sales in the last 3 years only on the established products, based on the established products, and therefore we are very, very positive on the now and the future of the established products. With GCAL in light blue, you can see that we are a little bit higher level of uncertainty built in, because this is a question of when and how fast the product will be taken on in which countries. I mentioned before, we are very positive about what happens currently in Sweden and other European countries to follow. But then there are big markets like US, etc. So it's a longer journey, and therefore as well, this is now a question of timing. Studies perform very well and results are promising, and the interest for this product is increasing every quarter. And of course, the highest level of uncertainty is our products in the product development. The worst case scenario is there would not turn out at all as a product, which, knock on wood, nobody hopes and believes. But the best is that very, very soon we can bring NT-proBNP to market, which we just don't know. We are very transparent in our quarterly reports as well about where we are. We achieved great progress with the development, but we are not yet at a stage where we could define a firm launch date. So all in all, 1 billion NOK if everything goes well, and the ambition to achieve a 40% long-term EBITDA margin. And if we look to the next slide, we can summarize again on the sales performance that, looking back, we achieved 28% compound growth rate throughout the last 4 years. And I would like to highlight the partners which we are allowed to mention in public, like Siemens, Beckman Coulter, and Roche. They belong to the top 5 in vitro diagnostic players in the market. We have all in all 5 of those partners, but and there, of course, there is a longer list of in vitro diagnostic companies out there. 2 of them don't want to be mentioned yet, which we of course respect. Next slide, please. Talking about the short-term achievements, when we look back what we have achieved until 2022, we continued our sales growth as well in Q1 23. We achieved a 53% performance with some support of currency, or organic growth was 40%, which is again quite encouraging. Our gross margin has been improved compared to the quarter in 2022, is 52% versus 44% in Q1. And with this, we achieved an almost EBITDA neutral performance. And as well as a highlight, we had several studies being published on GCAL, on our product and market development, again confirming the clinical relevance of this product. And as well, those results came from Sweden, but as well now from other countries. Next slide. We have spoken about the EBITDA margin and our profitability objectives. As a growth company, we are investing in R&D, but we expect as well from ourselves that our current sales growth is demonstrating leverage on our other cost level. And therefore, on the left graph, you can see the performance of our cost of goods and other OPEX between 2018 until Q1 23, and the trend is right. Important probably as well for you to know is that our revenue source is quite stable when you get a customer on board, direct customer, but as well, especially the commercial partners. These are long-term relationships. And so therefore, it's not that we have to start every quarter from zero. We know that there will be orders coming. They can vary depending on disease spread. In summer, you have a lower level of diseases, so there is some fluctuation, but we never start from scratch every quarter. And probably as well important to know is that our cash position by the end of Q1 was 76 million NOK. Next slide. All in all, to conclude, we believe, and I hope you agree with us, we have an attractive value proposition in place, which drives value for customers, but as well for shareholders. The market we are serving is growing, and we are not only counting on the market growth itself, we are as well having a strong commercial plan and talents in our company to enable as well competitive convergence. We are not a one product company. We have 3 product categories, all in all 7 products, of which 5 are already launched. We are on track with our profitability plans, and all in all, we believe for our shareholders, we are positioned for a strong value creation. And with this, I would like to finish and answer any kind of question.
Thank you very much, Hilja, for a good presentation. I do have some questions, and I think that the ProBNP is very interesting. And I'm wondering if that could be used to diagnose other cardiac diseases as well, beyond congestive heart failures. I guess, for example, valvular heart disease also expresses ProBNP. And is that something you could easily move into as well?
Yeah, first things first, you are right. And this is what you do throughout a life cycle of a product. For regulatory reasons, focus on the most established disease group, bring the product to market, demonstrate performance, gain traction in the market, and then start new studies performing again into other disease areas, which you mentioned, for example, could be great ideas.
You mentioned in the presentation that this was a longer term, or say longer duration R&D project. Would expanding it into, valvular heart disease also be challenging in terms of R&D, or is that more regulatory issues than anything else?
Yeah, so if you change the product, it would be a completely new product development, which I don't believe. Different disease areas, in my experience, which is more than 30 years, is that you find other disease areas, and we have that as example as well with GCAL, our calprotectin. It can be used in all kinds of disease settings. We focus currently on severe infections and mainly avoidance of sepsis, but with anti-ProBNP, we see exactly the possibilities when the product is developed to look into other disease areas, and that would be a regulatory point of work, not a product development point of view work.
Okay, perfect. And you mentioned the higher sensitivity in ProBNP as well. I mean, do you have any data on now how much earlier you could detect BNP presence than what's available now?
So the faster time to result is due to the shift of the assay from analyzers which take 2 hours, 3 hours, to an analyzer which takes now 10 minutes. So that is the difference in timings. The sensitivity level is already in place as well with the other assays in the market, but it has never been achieved on the clinical chemistry platforms. This requires a special immunoassay technology, which we master. And nobody has ever achieved the sensitivity level going so low with this technology. And that is why we have to innovate this well-established PTR technology from scratch and breaking the barrier here.
Okay, perfect. I think that's very interesting. Now, as a last question, I just wanted to ask a sort of basic one. I mean, what's the barriers for clinics to use your type of technology? Do they need to make any sort of investment or change processes, or is it more of a plug and play?
Fantastic question. I mentioned it very shortly in the beginning of the presentation. The beauty of our offer is that there is a very low barrier for change, as they can add, the laboratories can add our reagents on the established instruments they have already in the laboratory. And that is a very fast a process which we support as well with our own personnel and with our partner personnel. It's not to be compared if you would need to install an instrument and all the respective changes and protocols you have to follow there. So a very low barrier for gaining new customers.
Perfect. That sounds like a great setup. So thank you very much, Hilda. That was all the time we had today. We'll now go for a short pause and return with Ultimovacs in a few minutes. Thank you.
Thank you.
The next company presenting today is Ultimovacs, a Norwegian biotech company developing novel cancer vaccines. And with us, we have here today Carlos de Sousa, the CEO of the company. Carlos, the stage is yours.
Thanks, Gonzalo, and good morning, everybody. It's a pleasure for me to introduce you to Ultimovacs. As Gonzalo mentioned, we are a biotech located in Oslo and also with a team and an office in Uppsala. In the next slide, I have to show you because, as you probably know, we are listed in the Euronext Oslo Stock Exchange. And if we move to the next slide, then I will give you a very brief introduction to Ultimovacs, and then we'll tell a little bit in more detail about our clinical program and the exciting times really just around the corner for the company. So we are a clinical stage biotech company. We are developing a universal off-the-shelf cancer vaccine in quite a broad clinical program. Our vaccine targets telomerase, or basically re-educating system to kill cells expressing telomerase. And telomerase is expressed in 85 to 90% of all cancer types at different stages. In terms of the vaccine itself is easy to use and is intradermal injections and has the potential to be used in multiple cancer types. We have quite a broad clinical program. We are currently in 1 phase 1 and 5 phase 2 trials. And we also have multiple phase 1 long-term follow-up studies. All of them have shown a strong safety profile, efficacy signals, and also very importantly, a duration of the immune response. This means that, for instance, 8 to 10 years after these patients were vaccinated, we still find these immune cells that recognize telomerase. As I mentioned, we have very near-term value inflection points. As a matter of fact, now during June, we expect the readout of one of our phase 2 studies, followed by another one in the second half of 2023, and the third one in the first half of 2024. So within a period of 12 months, we will have 3 readouts from phase 2 studies, all of them randomized and comparative. The data that we have produced so far was submitted to the FDA, and we received a fast-track designation and orphan drug designation in metastatic melanoma. So this really provides validation from the FDA. Also, in our program, as you will see, we have several joint projects with key oncology groups, in the US, in Europe, and Australia, and also collaborations with large pharmaceutical companies. So all in all, we had a very exciting program, a very ambitious clinical trial program, really trying to show our goal that UV1, our vaccine, can be used across multiple cancer types and in combination with different checkpoint inhibitors that are now the leading class in treating cancer. So if we move to the next slide, just to briefly tell you why did we pick up telomerase. Telomerase is what is called one of the hallmarks of cancer. This is all the characteristics that normal cells need to express in order to become cancer cells. And telomerase, that is the mechanism that 85 to 90% of tumor types choose to give these cancer cells immortality so that they can divide without dying. So this means also that UV1 has the potential to be used, applied in a broad range of cancer types, as you also see part of the reflecting in our clinical program. Because the cancer cells need to express telomerase, so they cannot mutate out of telomerase, otherwise they basically just die as normal cells. This is a very important target, because even if the tumor mutates, and this is very common in cancer cells, they will not mutate out of telomerase. And because telomerase is expressed in the primary tumor, in the metastasis, from early stage to late stage, is a very good target. So if we move to the next slide, so let me give you a picture of our ambitious and broad phase 2 program. When concluded, we will be recruiting more than 670 patients. Melanoma skin cancer is our lead indication. You see there in the top, we have these 2 studies already completed, and I will show you a little bit of that data. And then you see also out of the 5 phase 2 studies, as I mentioned, all randomized comparative, we have completed enrollment of patients in the first 2 trials in melanoma and in mesothelioma. And we expect the data from the mesothelioma study now in June, and then from melanoma in the second half of 2023, followed by head and neck, ovarian cancer, and non-small cell lung cancer. So as you see, quite a broad program. And you see also that we collaborate with Bristol Myers Squibb in the mesothelioma study and with AstraZeneca in the ovarian cancer. So a very broad program, and that we hope will show the value and the impact that UV1 can have when added to the treatment with checkpoint inhibitors. We also have a second technology, an adjuvant platform that is now in phase 1 in prostate cancer. So if we move to the next slide, the first 2 phase 1 studies in combination with ipilimumab and pembrolizumab, that is Keytruda. Basically, these studies, although in small number of patients, showed that we have a good safety, very important in these sick patients and that have to be treated with multiple drugs. Also, a good time to progress, and also, an extended overall survival that is very important. We want these patients, after treated, to stay alive for as long as possible. This data was presented at different conferences and in publications, and as I mentioned to you, received the fast track designation, orphan drug designation. But let me show you on slide in the next one about the data from the combination of UV1 with pembrolizumab that is one of the standards of care in melanoma. What you see there, it looks like a very complex slide. It is a bit, but what you see there, the graph on the left, each line corresponds to one of the patients treated in the study. And each box corresponds to an image that was taken of this patient to assess the progression or regression of the tumor. So you see in green, these are all the patients that had a good response. Basically, they stop or reduce the size of the tumor. And in some cases, what we call a complete response, the tumor totally disappeared. And you see here, very important, and I want to emphasize, is that when the patients responded, is that this response stays, you see there, about 2 years follow-up, that when after the patients respond, they maintain this response for an extended period of time. The other part part of data that we also observed was that we had 10 out of 30 patients that had a complete response, means that they got rid of the tumor, and another 7 have a reduction of the tumor. This is unusual because normally you have more partial response than complete responses, so was not in this case. So this is very good for patients and really a reflection of our mechanism of action. The other part that was also very important finding is that normally there are some biomarkers that patients respond worst to the checkpoint inhibitors if they have what is called PD-L1 negative. And very good to see that in our study, it didn't matter. So this means really that we cover a broad group of patients, and this compares positively with the historical data. But of course, what we want to show is in bigger studies, as you see in the next slide, with more patients and comparative. This means that, for instance, in the initial study that is in melanoma skin cancer, this is first-line advanced melanoma, we have 2 arms. One arm is ipilimumab nivolumab, one of the standards of care, and then is compared with a second arm that is ipilimumab nivolumab plus UV1, our vaccine. So then in this way, you have the patients in the same study and you can compare the efficacy and safety between these 2 arms. We concluded recruitment of these patients basically in 2 years. We concluded June last year, all during the pandemic, and we were expecting the top-line result the first half of the year. And this is reached when 70 patients have progressed or died, and very positive for patients. It's taking them longer than expected to progress or die. So we had to change our guidance to the second half of 2023. And the study ran, basically half of the patients in Europe and half of the patients in the US. So the next study, the NIPU study, mesothelioma, very, very hard to treat cancer. Patients progress very fast. Mortality is very high. And is also a combination with ipilimumab, nivolumab, plus UV1. And this is the study that we also completed the enrollment, and we expect now top-line results during June, so really just around the corner. And the study ran in Europe and Australia. And then the third study that we expect data, and that will be in the first half of next year, is in head and neck cancer, another very tough cancer to treat, very low efficacy from pembrolizumab. It is a standard of care. And the study all ran in Germany is now more than 80% recruited. And as I mentioned, we expect data in the first half of 2024. And then more recent studies in ovarian cancer and in different European countries, and lung vac in non-small cell lung cancer in Norway. And these studies, are early stages of recruitment, but now moving quite nicely. So very ambitious program, and we hope that we'll be able to show that UV1 has a potential to be used across the board of multiple cancer types and in combination with different checkpoint inhibitors, that is depending on which ones are used in each of these tumor types. So if we move to the next slide, I want to tell you a little bit of the key benefits and in the very competitive profile of UV1. And the fact that it is a easy to use product and with low production costs and simple logistics make it very accessible for patients to be treated regardless of where we are. What we call an off-the-shelf product means that the UV1 is ready to be administered when needed. You don't need to do any screening of the patients before, you don't need to do any biopsies. The product is ready to be administered when needed. And it's just treated with 8 intradermal injections, so no hospital complex infrastructure required. So patients really can be treated regardless of any way where they are in community centers or main hospitals. The fact that it's a simple manufacturing is also very important because this is when we produce product that we'll have now, by the end of this year, what we call a commercial process in place, is very important that it's easy and low cost. And as I said, the fact that it just sits in the normal fridge and ready to be administered when needed makes it, all of these characteristics a very important profile. So if we move to the next slide, let's talk a little bit about what is the potential for UV1, if we have positive data. As I mentioned, we will be combining with checkpoint inhibitors. This is the lead class in all of pharma, with quite a healthy growth rate, 11% CAGR. And as you see there, basically we are combining with 5 out of the top 6 checkpoint inhibitors currently approved and on the market. So this really shows the potential for UV1 to grow in different indications, and also, for instance, in moving to early stages of the disease, what we call a neoadjuvant phase. So even a small share of this huge market, close to $70 billion annually in sales, it's very important and a huge potential for UV1. So if we move to the next slide, just to give you just a little bit of information on our financial situation that is quite good. It's very important for the company to be solidly financed. We are, we did an IPO in the Euronext Stock Exchange in 2019. After that, we did 2 additional private placements, both of them oversubscribed. And I think it's important that for a company that has 26 employees, both in Oslo and Uppsala, we have quite a very healthy market cap, about 4.3 billion Norwegian krone, approximately $410 million. And we have a cash position of approximately 406 million Norwegian krone that, as I said, very important, gives us an estimated financial runway to mid-2024 and takes us across the results from 3 of these studies. We have quite a solid group of owners with long-term perspective, and this lets the team focus really on the business. These are primarily big family funds, but also the Norwegian pension funds, some additional institutions, and we have our goal to basically expand the number of shareholders and the geography to other European countries and the US. So if we move to the next slide, just to really give you a perspective, what is coming. As I mentioned, very exciting times for the company. You see there the NIPU study, as I mentioned, expected now to have the readouts during June. Then the second half, the Initium study, and then in the first half of 2024, the focus study in head and neck. So, all of this data is extremely important, can be transformational for the company. If we have positive data, this will be very important to show that the cancer vaccine concept works. Cancer vaccines are now much more visible and of interest for a big audience after some data from other cancer vaccines. But we believe with our competitive program, we will have a very good place in terms of further development. We will be discussing with the authorities how we move forward with UV1, but we will also, with positive data, be discussing with strategic partners the possibility to enter into a partnership with a big pharmaceutical company. As a company, we are prepared to move forward, but we believe that a large pharmaceutical company with more financial resources can bring more indications to the market and, of course, help more patients, more cancer patients to be treated. So again, we will be keeping you informed that these studies progress, so please keep aware of our news. So in summary, just to finalize in the next slide, as a company, we operate in a very exciting field, the immuno-oncology, with cancer vaccines that are universal, easy to use, and broadly applicable in multiple cancer types and in multiple combinations. It's an off-the-shelf, and as I said, easy to use, the profile, and very broad clinical program, and that has the potential to show the extended use that UV1 can have in multiple cancer types. We expect readouts from 3 studies within the next 12 months. We have external validation from regulatory authorities and joint projects with large pharma companies, oncology specialist groups. We have an experienced team, a strong shareholder base, really supporting the company. And again, very exciting period of time for the company. And I want to thank you now for your attention, and I'm open for any Q&A. So the word to you, Gonzalo.
Thank you very much, Carlos. Very nice presentation. A few minutes for questions now. And as you know, ASCO is about to start. And last week, when the abstracts got released, one abstract from Regeneron, where they are combining cemiplimab with fianlimab, in first-line melanoma, they showed quite interesting data from a phase 1, of course, but they showed an overall response rate of 63% and a PFS of 14.7 months. And I was wondering, if this is validated in a phase 3, that could represent a threat to ipi/nivo first-line in melanoma. And I was wondering if you could give us some color on your thoughts around this, and how do you see the space of LAG-3 inhibitors progressing?
I think cancer patients need as many treatment alternatives as possible. I think we have been after the Optulwag, that is another nivo plus LAG-3 combination. We also spoke with KOLs, and they see there is a room for that combination, but because of the efficacy not being at the level of ipi/nivo, they still prefer to use ipi/nivo, and they see more that the LAG-3 anti-PD-1 combinations will be probably replacing the monotherapies. For us, it really doesn't matter. You know, if we have good results with ipi/nivo, basically, you know, the mechanism is the same if you combine with a LAG-3 or and the anti-PD-1. So we see that as also another opportunity for UV1 growth into the treatment class. So, you know, again, we want to see as many treatment options for cancer patients, but we don't see that as a threat for us. We see more as an opportunity to have further combinations.
Great, thank you very much. A second question is on this split of results that you have in between PD-L1 positive and PD-L1 negative patients. I was wondering if you could give us some thoughts on what would be the ideal phase 3 trial design. Would you go for both populations, and if you go for both, would you power the study to score significant results in both groups independently?
No, it's an excellent question. You know, I don't really have an answer to give you, because this will be part of based on the results we'll have from the current studies and discussion with the authorities. We think that shows a potential that we want to explore further. You know, there are indications, for instance, like in non-small cell lung cancer, where there is a clear separation of the treatment alternatives for patients, depending if they have a high PD-L1 expression or a low PD-L1 expression. If the patient has a low PD-L1 expression, you know, the checkpoint inhibitor needs to be combined with chemotherapy. So, for instance, this will be an area if we show that we reduce the need for chemotherapy, that will be very beneficial for patients. So it was a very exciting finding, you know, that we see that we want to explore, and you know, we'll be having discussions with KOLs and authorities what will be the best way of exploring that potential.
Thank you very much. And the next question is on your TED platform. In the second half of the year, you're planning to announce top-line data of the study you have conducted. And I was wondering if you could give us some words on what is the data that you are planning to release, how much can investors expect from this data, and about the future plans after that.
You know, the data, this is, again, a phase 1 study that was primarily to assess safety, safety, you know, we were also looking at immune activation, but this is just one piece of what we are doing with that. We have multiple non-clinical or non-clinical studies, you know, running, also CMC activities. So we expect, you know, in the second half of the year, you know, later in the year, to be able to provide the, you know, results not only from TENDU, but also from all of these non-clinical activities we are performing, and what will be our expectations for the overall TED platform to be moving forward. So, you know, will be broader than just the TENDU study results, because this is just a small component. We will keep the market updated, definitely.
Mm-hmm. Thank you very much. And one final question, it's on your NIPU trial, the mesothelioma trial. If I recall correctly, is an investigator-initiated trial. And I was wondering, what are the implications in terms of data handling if the trial is investigator-initiated, and what are the implications also for a potential phase 3?
You know, that's, you know, all these studies, you know, you have, you know, different types of investigator-initiated studies. You have very small studies, and you have, you know, large studies that basically all of our investigator-initiated studies are quite a big number of patients. And I think the important part there is that the protocol and the statistics are the standard for any phase 2 program. So it doesn't matter in this case if it's an investigator-initiated study or a company-based. This data will always be supportive, and any phase 2 study, the major value is to show you what are the patients and the type of design that will be ideal to move into phase 3. So if the data is positive, we'll of course be sharing that data with the authorities and discuss what will be the next step, you know, for a phase 3 in mesothelioma. That, as you know, is a very, very tough cancer, and you know, the current treatments are really not that great. So these patients need as many alternatives as possible. So definitely, in the back of positive data, we will have very open discussions with the authorities.
Great. Carlos, thank you very much for being here with us today and for your presentation.
Thanks, Gonzalo. Thanks, and thank you, everybody.
Next, we have Hubro Therapeutics, a Norwegian biotech company developing cancer vaccines with a focus on frameshift mutations. And with us, we have here today the CEO of the company, Jon Amund Eriksen. Jon Amund, the floor is yours.
Okay, good morning, everyone. It's a pleasure for me to present Hubro Therapeutics to you all. And maybe you can go to the first slide. Yes, as already mentioned, we are a Norwegian private clinical stage biotech company, and we develop vaccines and also diagnostic tools targeting the frameshift mutations that occurs in genetic microsatellite instability cancers. And the company is about 4.5 years, but our technology is based on research of the last 30 years or so, mainly at the Radium Hospital in Norway, but also at by several companies. And we, as said, develop therapeutic and prophylactic cancer vaccines along with companion diagnostics. So our, I would say, vision and mission is to both detect, protect, and follow up and treat cancers. So we develop diagnostic tools that are able to detect the frameshift we are targeting in liquid biopsies. And our therapeutic vaccines are for treatment of MSI cancers. We have 2 vaccines in development. And the lead candidate vaccine is FMPV1 targeting colorectal cancer and gastric cancer. And also we develop a vaccine, FMPV3, for treatment of progression of cancer in high-risk groups like Lynch syndrome. And also our diagnostics tools can be used not only to detect the frameshift mutations, but also to follow up after treatment. So we can monitor effective treatment, and also after prophylactic vaccination, we can detect early progression of cancer. So if we go to the next slide, frameshift mutations, they are genetic mutations. They are caused by mismatch repair defects in cells. So if mutations are ongoing in cells all the time, but are usually repaired by the DNA mismatch repair system. So in certain cells, you have a mismatch in or a deficiency in the mismatch repair system. And then so mutations are not repaired, and they become somatic mutations. And that will create a shift regarding frameshift mutation, will create a shift in the protein synthesis that again will create neoantigens that are perfect targets for vaccination. So a frameshift mutation occurs in microsatellites in the genes simply by deletion or insertion of a nucleotide in a mononucleotide repeat like you can see in the picture here. And then if one nucleotide is deleted, you will have this shift, and then after the mutation, you will have a completely new protein sequence or a neoantigen that are highly specific for the cells in which it occurs. And these neoantigens are perfect for targeting by a vaccine. And the next slide. It's also very important to select the right types of neoantigens for a vaccine. And we develop off-the-shelf vaccines targeting shared neoantigens because we think that's the best way to approach treatment of cancer with a vaccine. And the shared neoantigens, they are perfect because they have a high tumor specificity, there's no central tolerance, and also it's a high prevalence in the patients, like demonstrated in the table here. And regarding vaccine designs, there are several types of mutation in a tumor. And the better one to target with the immune therapy is, of course, the early occurring mutations or the driver mutations, because only targeting these mutations can really have a long-term effect on the tumor, because the later occurring local clone mutation If you target those, that will only prune the cancer, and the cancer will be very quickly repopulated by other clones, cancer cell clones. So we target frameshift mutation in TGF-beta-R2, ASDE1, and TAF1-beta. They are truncal and or early occurring mutations commonly present in MSI cancers, both sporadic and hereditary, stemming from Lynch syndrome. And as you can see from the table, the prevalence in the different cancer types are quite high, and that makes them ideal for a off-the-shelf vaccine. And we use peptides, and of course we use long synthetic peptides because it's very important to induce both CD4 T helper cells and also CD8 T cells, cytotoxic T cells. And the reason for using the long peptides is that we can have a natural representative antigen processing of the vaccine. And we can also, it's easy to produce the peptides, and the peptides are stable and it's suitable for off-the-shelf vaccines. On the next slide, please. So based on our technology, we can develop several products. Recently, we have just finished the phase 1 study with FMPV1, which is a peptide, therapeutic peptide vaccine targeting colorectal and gastric cancer. And we have finished phase or preclinical work with FMPV2 targeting ASDE1, which is quite prevalent in endometrial cancer. And we are also developing GM-CSF, which we use as adjuvant with the peptides. It's important to use a good adjuvant because the peptides themselves are not very immunogenic. And the prophylactic vaccine FMPV3 contains 3 different antigens, and the 3 neoantigens are representative for MSI cancers developing from Lynch syndrome. And in addition, we are also working on the diagnostic test targeting the same 3 frame shifts. Next slide, please. So in the therapeutic setting, there are 3 big MSI cancers, actually, and that's colorectal cancer, gastric cancer, and endometrial cancers. And as said already, we develop FMPV1 against colorectal and gastric MSI cancers. And if you look at the funnel there, you can see that if we correct or calculate the prevalence of MSI cancer and prevalence of the frameshift mutations, you see that we still have quite a significant market for the therapeutic vaccines, something around 200,000 annual incidences of these MSI high cancers. And the next slide, please. And also in Lynch syndrome, which is the target for prophylactic vaccination. The Lynch syndrome is a high-risk group, and it's a genetic disorder where you inherit the mismatch repair deficiency for your parents. So because of that, you have a lifetime high risk of developing MSI high cancers. So as you can see, it's for some certain patients groups or subtypes of Lynch syndrome, you have up to 90% risk of developing some kind of MSI cancer during your lifetime. And it's also a very high risk for developing MSI colorectal cancer and endometrial cancer. And the prevalence of Lynch syndrome in the population is quite high. It has been much more focused on Lynch syndrome recently, and that the prevalence is increasing. And it's been documented that about 1 in 279 subjects actually have Lynch syndrome and have a high lifetime risk of developing cancer. So, yes, next slide, please. And the mechanism of action is that it's quite simple. It's that you inject GM-CSF and the peptides intradermally in the skin. So it's a local administration. And then the peptides are taken up by activated dendritic cells. and travel to the lymph nodes where they present the peptide epitopes after antigen processing of the vaccine peptide. And then they induce T cells that are specific for the neoantigen. And the T cell enters the circulation and home to the tumor, where the T helper cells, that's very important to induce T helper cells because they are not recognizing the cancer cells in the first instance. They are reactivated by dendritic cells in the tumor. So reactivated T helper cells, they actually facilitate the full attack on the on the tumor cells by CD8 cytotoxic cells, by producing pro-inflammatory cytokines, and also by cross-presentation of new cancer neoantigens. So after one cycle, you will have a broader immune attack on the cancer cell because of cross-presentation and activation of CD8 cells recognizing also other tumor antigens. And it's very important to mention that in the therapeutic setting, we will have to use the vaccines together with a checkpoint blockade. Because as you probably know that T cells attacking the tumor cells, they will very quickly be deactivated by interaction between receptors on the T cells and protective ligands on the cancer cells. So checkpoint blockade actually take away that break and reactivates the T cells. So next slide, please. Yes, we have just completed a phase 1 study with the FMPV1 in healthy volunteers. And the objectives for that was, of course, to study tolerance and safety and, of course, immune response, because we really wanted to assess the immunological potential of the vaccine before we go into patients. So, and the results were more or less like we expected. The safety was fine and great tolerance. And the only thing we saw was the normal injection site reaction that you always see when you inject something in the skin. And the immune response, we detected immune response in 15 of the 16 treated subjects after 4 vaccinations, 4 weekly vaccinations. And the DTH test is a skin test that you do in vivo in the subjects by injecting the antigen, and then you get a skin reaction if new antigen-specific T cells has been induced. And you can see an example of a positive DTH reaction on the picture on the slide. And of course, all the subjects were DTH negative at baseline before vaccination. And we are also following up by doing T cell proliferation assays. So we have been able to detect T cells in blood that are neoantigen or vaccine specific. And that increased after baseline and after vaccination. So all in all, this phase 1 study was quite successful, and it paved the way for a phase 2 study that we now are planning. Next slide, please. Next slide, please. Yes, and we are also, was that the next slide, or did you skip one? Yeah, there it is. And as said, we are planning a phase 2 study in metastatic MSI colorectal cancer. And that study will be done in combination with standard of care checkpoint blockade. And the study design is a Simon 2-stage algorithm, and we need around 56 patients, we believe, to meet to reach the objectives, so we can have a proper readout. And it's also an interim readout after 20 patients, where we can see and get an indication how it goes, so we can either have the opportunity to increase the dose or even to amend the study protocol if needed. And we submitted the CTA in the UK, where the study will start. It will have a 2x3 patient lead-in, simply because we need to assess the compatibility of the vaccine together with standard of care CPI. That hasn't been done before. So we need that safety lead-in cohort. The CTA was submitted actually in December last year, and we haven't got a response from MHRA yet. And the reason for that is not our reason, but not caused by us, but it's because MHRA have a significant backlog because of the pandemic. So we expect to have the feedback at least within a couple of weeks from now. At least that was the promise of. So yeah, I think it's reasonable to believe that we will start the study in late in Q3 or early Q4 this year. Okay. Next slide, please. And we are also contemplating to do a phase 1 study in healthy volunteers again to demonstrate the safety and immunogenicity of the 2 other vaccines that we have completed the preclinical work for. And that at the moment we are producing the vaccine, and we hope to start the study early 2025. Next slide, please. And also we develop diagnostic tests that we think might simplify the procedure for diagnosing MSI. Today that is done by using biopsies, physical biopsies, and a lot of sequencing, the immunohistochemistry and so on. It's quite a tedious procedure. Our test can be used to detect cell-free DNA from liquid biopsies. It's a very simple and easy procedure. It's cost efficient. And also, very importantly, a liquid biopsy is a global presentation of the tumor. And a physical biopsy is only a local presentation of what's in the tumor. So we believe that we have a very good test that we can also use to detect or identify patients that we are going to treat, and also to monitor effect of treatment, as said, and also to diagnose MSI. And it's an easy test. It's a PCR-based test, and we have our proprietary designed primers that we use. So we don't need sequencing. We can directly detect the frame shift, for instance, on a gel after electrophoresis. That will cut down the time for doing the analytical work and also cut down the cost for doing that. And the next slide, please. That's a picture of what we see as competition in our field. It's not for oncology in total. It's basically for immunotherapy of cancers. And as usual, it's a somewhat busy picture regarding colorectal cancer. It's a lot of activity there, of course. And it's led by Merck that is continuing to try to expand the market for their checkpoint inhibitors. And we are, as said, also in phase 2, and we will work together with checkpoint inhibition. So that's not really a competition for us as we see it. It's more like we see it as a potential collaborator for the future. And also in gastric cancer, the picture is quite similar to colorectal cancer. And in endometrial cancer, we are quite alone regarding immunotherapy. And in Lynch syndrome and prophylactic vaccination, it's Hubro Therapeutics, and it's another company called Nuscom. It's a Swiss company that develops a virus-based vaccine. The technology between the 2 companies are quite different. We target a few very important neoantigens and frameshift mutation, while NuScomp targets, I think they have a virus containing something like 209 potential cancer neoantigens. And that's a very different technology and strategy. So what we know, at least, is that when we see an immune response, we know it's a correct immune response. And the next slide. That's simply an overview of our IP. And we are a young company, so our IP is also quite new. We started to file a patent application in 2019. And the first one was to cover TGF-beta R2 and FMPV1. It also contains not only the peptides, but also the nucleotides, T cells, and composition of vaccines. So, and that is the most advanced application, and we have now received the notice of intention to grant from EPO. So that's quite encouraging for us. And we also have a second peptide vaccine application. That's the second one there. And that's covering the other antigens we are developing, as well as other formulations and cocktails and T cells and composition again. And we have also filed patent applications for our designed primers and diagnostic tests that are still pending, of course. And then next slide. That's our team. We have a very experienced and competent team, and we also are strongly supported by our experienced board of directors. So yes, next slide is probably the end of the story. So thank you all for listening.
Thank you very much, Jon Amund, for the presentation. Very nice. We have a few minutes for questions, and I will start asking you about this pack that you could bring to the market of the vaccine together with a diagnostic test. How easy or hard is to make it into the market for these tests? Do you have to be like included in some kind of guidelines, like country-based guidelines in Europe? And do you see also potential market uptake of the vaccine without the test being approved?
I think we both ways. I think basically we see the opportunity of developing the diagnostic test as companion diagnostics, but we are not sure if we need it as companion diagnostic because the prevalence is very high. And we will, of course, continue to monitor also the prevalence of the frameshift when we treat the patients. So it might be that we don't need it as a companion diagnostic for the vaccines. But in any case, we wish to develop the test for at least for in vitro diagnosing because it can be used to follow up and also for detect and monitor effect of treatment in the prophylactic setting because that's we think that as it looks where we are in development at the moment, the tests are very, very sensitive. So we hope that we will be able to use the test to diagnose Lynch syndrome subjects for an early detection of cancer so we can get them very early into treatment.
Great, thank you very much. A second question is on this competitor that you mentioned, NuScomp. They have a different approach or a different technology, let's say, to design their neoepitope vaccines. You mentioned that they go for above 200 neoantigens in their candidate. You will go for way less. Could you give us some color on the differences of going for more like targeted neoantigens and potentially more prevalent or immunogenic rather than going for a broad panel of neoantigens like NuScomp does?
Yeah, I think for me, it's basically the science and immunology here, because when you inject a vaccine, it will be taken up by dendritic cells, and it will undergo antigen processing. And the antigen processing is quite a selective process. So it's not so that you can expect that you will see immune responses to everything that you inject, because it is a selective process. So only the the peptide epitopes with the highest affinity will actually be presented to T cells in HLA molecules. And that's why we target only 3 antigens that we know are crucial to the tumor in very early occurring mutations. So we are simply trying to cut off the tumors by the root. And if you, like say, have a haystack of neoantigens, it's not so that you can expect to see efficient immune responses or T cells against more than maybe a handful. Because when you see immune responses to a virus, it might be only 2, 3 epitopes that really are the protective immune response. So that, I think that's the big difference between our technologies. We try to provide the immune system with the needle in the haystack, if you can say it that way.
Great, thank you, very clear. And one last question. You're about to start or planning to start your phase 2 soon. I was wondering if you could give us some words on your cash position now and how well covered you are.
Oh, we are in the process of raising money at the moment. So and we need to raise money to really to support the study.
Great.
That's exactly where we are at the moment.
Very clear. Thank you very much, Jan, for your presentation and for being with us today.
Okay. Thank you. Bye-bye.
The next company presenting today is Lytix Biopharma, a company, Swedish biotech, Norwegian biotech, sorry, developing oncolytic peptides for the treatment of cancer. And with us, we have here today the CEO of the company, Øystein Rekdal. Øystein, the floor is yours.
Thank you. So thank you for this invitation and opportunity to present our new class of immuno-oncology drugs and how these drugs can address the major challenge in cancer treatment. So cancer is still one of the largest health programs globally, with more than 10 million cancer-related deaths in 2020, and this number is expected to increase during the next decades. So clearly, there is still need for better treatments. One new pillar in cancer treatment has revolutionized the cancer treatments the last decade, which is immunotherapy. And immunotherapy target the immune system, the patient's own immune system, rather than target the cancer cells. And with some superior effects in certain cancer types with immunotherapy, led by immune checkpoint inhibitors, the global market has increased significantly for this new pillar. So within this new pillar, we are developing a new class of immunotherapeutic drugs, which may address some of the major challenges. It has already been validated by international leading experts in the field. We have a strong presence in the US, we have a strong advisory board, and we already have in place a licensing agreement with a US company. So our drugs has the potential to be used for a number of cancer indications, and our lead candidate is currently in phase 2, several phase 2 studies, so therefore we are seeking for additional partnership in the same time. So since immunotherapy is also predicted to grow significantly the next decade, this decade, and our drugs has the potential to be used for a number of cancer indications and address the major problem in current treatment, there is also a large commercial potential for our drugs. So even though immunotherapy has shown some superior effects in certain cancer indications, why is it still so difficult to cure cancer? The major reason is that cancer cells mutate over time, resulting in a number of different cancer cell variants in a solid tumor. And since there occurs unique mutation in each tumor, there are no tumor are similar, and we get intratumoral and intertumor metastatic and interpatient heterogeneity. And some of these cancer cell variants either are or become resistant to current therapy, making it extremely difficult to cure and eliminate all the cancer cells in a patient. And failure to do so results often in an even harder to treat tumor, resulting in few options for the clinician and a sinister prognosis for the patient. So that tumor heterogeneity is the major challenge, is illustrated or supported by this survey done among clinical oncologists, which ranked heterogeneity as the major problem in current therapy. So the question is it possible, since the immune system are able to recognize many different antigens among viruses and bacteria, is also able to be mobilized to recognize all the different tumor antigens or mutations in cancer? We have, by giving our lead candidate locally in a tumor, shown that LTX-315 is able to mobilize the patient's own immune system to target distant, non-treated lesion in patients. As illustrated with the sarcoma patient to the left, where we treated one lesion in the lower gluteal muscle, resulting in a 63% size reduction of a lesion in the lung. Another melanoma patient were treated with in 2 tumors on the right side, and resulting in a reduction of to up to 82% of one lesion and 39% reduction of another lesion, indicating that 315 is able to mobilize the immune system to target distant non-treated lesion. So the immune cells that are important for killing cancer cells in our body are T cells. And we have shown and demonstrated that LTX-315 is able to significantly increase a number of different T cells in the patients. Illustrated here by this breast cancer patient, where we found that 126 different T cell clones were significantly expanding in the patient. And these T cells are able to reduce other non-treated lesion in the patient, illustrated with the waterfall plot on the left, where each column is representing one change in one lesion in each patient, showing a reduction of size up to 80%. And 1/3 of the patients so far has shown this systemic effect on non-treated lesion. So how does 315 generate this broad T cell response in a patient? So first of all, we tested LTX-315 against both drug-sensitive and drug-resistant cancer cell lines, including cancer cell lines that were resistant to multiple chemotherapies, and showing that it was equally active against the resistant cancer cell lines and drug-sensitive. And the reason why it can kill resistant cancer cells is the way it opens up the cancer cells and thereby solves the challenge with tumor heterogeneity. We have shown that our lead candidate is internalized and open up all the compartments in the cancer cells, releasing both tumor antigens and immune stimulatory molecules, which then evoke the immune system in a very powerful way. So this may be the reason why LTX-315 are able to work in therapy-resistant cancer types. So in collaboration with Harvard, we showed that in a BRAF-mutated melanoma that was resistant to chemotherapy and immune checkpoint inhibitors, it caught quite attention when our lead candidate was able to cure all the animals in this model. This photo to the right showed the control with a growing tumor, and after LTX-315 in the lower photo, a total eradication of the tumor. And the survival curve to the right showed that all animals survived after LTX-315 treatment, in contrast to the controls. So our molecules kill cancer cells in a way that the tumor antigens are exposed, and it also kickstarts the immune system. So the released mutations or tumor antigens are transported to the lymph nodes and presented for the T cells. The T cells that recognize these tumor antigens are activated and enter into circulation and look for cancer cells which have the same tumor antigens it has responded to. So even though immune checkpoint, as mentioned, has revolutionized cancer therapy, if you look at these numbers, you still see that the majority of cancer patients do not respond to monotherapy of immune checkpoint inhibitors. There have been some improvements in combinations, but still the majority is not responding. And the major reason for that is that immune checkpoint inhibitors need an active immune system to work, called hot tumors. And the majority of cancer patients have more cold tumors. To the right, you can see how our molecules are turning cold to hot, showing many T cells by these brown dots in the cancer. And this has been confirmed in 86% of the patients treated so far, that we increase the number of T cells in the patients, and therefore should increase the response to immune checkpoint inhibitors and make them more responsive. So we already have tested combination of LTX-315 and immune checkpoint inhibitors in some small studies, but there have been very promising signals where we have seen effects with 315 and anti-PD-1 inhibitor pembrolizumab, obtained effects not obtained with pembrolizumab alone. In this triple-negative breast cancer patient, that is the most aggressive type of breast cancer, we saw a reduction of tumor size up to 91% in the liver, the metastasis in the liver. the checkpoint inhibitor pembrolizumab alone have, in a big Keynote 86 study, shown no effect against the liver metastasis, whereas 315 and pembrolizumab show this effect. So this shows that there is a potential for synergy in cancer with checkpoint inhibitor. So therefore, currently we are running a combination study with LTX-315 and pembrolizumab in melanoma patients. And the cancer population is patient that has already failed on anti-PD-1 or PD-L1 immune checkpoint therapy, meaning that all effects found in this study can be attributed to LTX-315. The number of patients we expect to be completed mid this year, and we also plan to present some interim readout in the fall 2023. We are doing this study in both in major hospitals in US and in Europe. So our technology has been validated by strong international recognized immuno-oncology expert, including the Nobel Prize winner and the founder of modern immunotherapy and founder of the first immune checkpoint inhibitor, Jim Allison, which is also a member of our advisory board. We already have a commercial deal in place with Verrica, which has licensed LTX-315 for certain skin cancer types. We have a contract that gives us regulatory and sales milestones at more than $100 million and royalty rates at the double-digit level. We also have financial support in US through our anchor investor in the last placement, capital raise, PBM Capital, who has shown quite strong track record and building biotech companies into successful companies, commercial success. So looking on our pipeline show the strength of our platform with the ability to generate a number of molecules for a range of cancer indication. LTX-315, as mentioned, are tested in 2 ongoing phase 2 studies, one run by Verrica in basal cell carcinoma. We have already completed a phase 2 study in combination with adoptive T-cell therapy. We have a second generation molecule that are shown very promising effect in liver cancer and is at preclinical stage. And to broaden our pipeline and looking into the future, we are also preparing for the future. We are also a number of additional molecules in our discovery phase. Our molecules has the ability to work both alone and in combination, and has the potential for a number of cancer indication, and therefore a relative good potential prospects. So Verrica is running a phase 2 studies in basal cell carcinoma. They have completed the part 1 and already reported some interesting signs of effects. They are currently running the part 2 of these part 3 parts of the study, and we are guided that they will start the part 3 in the first half of year in 2024. The major treatment of basal cell carcinoma is surgery, which is quite painful and quite invasive. So 315 may represent a non-surgical option for patients suffering from skin cancer. BCC is the most common skin cancer, with approximately 3 to 4 million patients diagnosed each year in US, which represents a huge commercial potential. Our next generation molecule has shown superior effects in liver cancer and address some highly medical need in this cancer indication, liver cancer. In addition, it can be used for other deep-seated cancer types. So our task now is to really generate solid clinical data and also expand internationally through additional partnership. We have experienced management to fulfill that. In the lower part, we have Graham Curry and Stephen Worsley, our CDO and CBO, located in US. Graham Curry has had key roles in the development of 8 approved drugs. Stephen Worsley has more than 25 years experience with BD leadership. If you go to Baldur Sveinbjørnsø, or CSO, he has his scientific background from University of Tromsø and Karolinska. Gry Stensod has her experience for one of the successful companies in Norway, Photocure, that has brought their technology all the way to market. Jess Breisteins, our CFO, has his background from PwC. We also have a very experienced healthcare and capital market expertise within the board, chaired by Marie Roscoe, who has extensive experience in clinical development, in leader position within cancer treatment, cancer therapies, including cancer vaccines. She also has experience from the investment side, so really a complete chairman on our board. We have 2 members from Sweden, Evelina Vågersjö and Marie-Louise Fjellskog, who both are experienced within immuno-oncology. Marie-Louise Fjellskog is currently a CMO in a Boston-based company, but is commuting to some additional tasks in Sweden. Kjetil Hesdal has been the CEO in Photocure and led this company to be the major leading bladder cancer company internationally. Jason Rieger is the representative from PBM Capital and has a very strong experience in both scientific development and business development. Brynja Forgeskog is one of our major shareholders, has built a small transport service to being the largest Nordic transport service providers in the Nordic. So, seeing the prediction of this market for immuno-oncology growing significantly the next decades, our molecules plan to be combined with immune checkpoint inhibitors, also have therefore a potential because it addresses the major challenge and can be used in several cancer indications. And LTX-315 also has a huge commercial potential in basal cell carcinoma, and we are encouraged by the movement or the process in Verrica Pharmaceuticals and are looking forward to receive new information from them. We also are looking forward to present some interim data in a very important study ongoing with the 315 in combination with immune checkpoint inhibitors, and looking forward to a very exciting year and a half year to come. Thank you for your attention.
Thank you very much, Øystein, for the presentation. Very nice. We have a few minutes for questions now. I will start asking you, I think that you didn't mention anything on that, about the mechanism of action of your drug. Could you give us some words on that?
Yeah. So our drug is developed from a natural occurring class of peptides found in nature called host defense peptide. The first host defense peptide was in fact discovered here in Stockholm. And these molecules has a highly net positive charge. They are amphipathic, meaning they have the positive charge on one side, and therefore they are ideal to electrostatic interact with negatively charged membranes. Okay. Bacteria has that, but also cancer cells compared to normal cells are more negatively charged. And it has been proven that this class of molecules interact with these negatively charged compounds on cancer cells, which are only found on the inner membrane of normal cells. But also this ability to go into the cancer cells and open up the whole cancer cells, so all these tumor antigens and mutation are exposed for the immune system. This is very different from many of the chemotherapy who kill cancer cells in a way that it more shrink rather than open up for the tumor antigens. And I think that's the very important difference. But also we see, maybe a bit surprising, how 315 indirectly and directly activate some very important immune cells that bring these mutations and tumor antigens to the lymph nodes.
Yeah, no, very interesting, very interesting. And do you see, you say that tumors are negatively charged in general, but also red blood cells are negatively charged. Do you see any kind of interaction between the
No, we have tested that, and if I had time, I had time, this table. On the lower part, we saw that the red blood cells are not affected at all. So a much higher dose is needed to kill red blood cells.
Oh, very interesting. I have a second question on the trial that you're running with Verrica in basal cell carcinoma. I mean, right now the standard of care for this, as you mentioned, is surgical removal, and this actually works. I mean, in terms of efficacy, it has been proved that it works. What is the value that you can bring with 315 apart from like the surgical pain?
Yeah, that's a good question, because it works, as you say. You can remove it by surgery. Two problems. Well, all these places where BCC, basal cell carcinoma, are in sun-exposed skin, so face, hands, etc. So what you often end up with after a surgery, you get some scar, you know, and when new lesions are coming up in other places, you continue to make scars. And we have done some survey. I mean, no patients, especially US, where it's high, hate to do this. We talk with surgery, and so we maybe heal the wound better, but most importantly is that you activate the immune system, so you can protect against new lesions in coming up. I think that's the major advantage of our drug.
Interesting. And if we move to the melanoma setting, you are going for second line, let's say, or patients that progress from a first line treated with checkpoint inhibitors. In the second line is with pembrolizumab, where you are combining your 315. What are your thoughts in this checkpoint inhibitor re-challenge that you are doing now, like first line progressing to second line?
Yeah, I think we have taken the most challenging population to test it, but we believe this strong ability to activate the T cells, making the immune checkpoint response better, make this a very strong case if we succeed to get the strong signal.
Yeah.
So I think, and the good thing is that you can do relatively small studies because they have failed on the immune checkpoint inhibitors. So all effects you got with the checkpoint inhibitors can be due to 315. If you go earlier stage, you have to have a higher number of patients. Of course, it's challenging. And I think melanoma, we are doing this in melanoma because that's the most immunogenic tumor type where everybody tests. But let's see this signal, get a clear signal, and probably we may expand with an additional patient to really get the significance, statistic significance. And of course, all immunotherapy want to move to earlier stage because these are sick, they have exhausted immune system, so you really are looking into the most challenging place. But if you do get something signal here, we have a lot of good opportunities.
Do you think 315 has a place also in the first line?
Well, I think that many patients do fail on immune checkpoint inhibitor because, and we know this, they lack T cells. So I think a compound, and I believe strongly a compound that can boost the T cells will get many more patients to respond. So I think a combination of immune checkpoint and a compound like our drug that activates T cells should be on first line.
Great. And one last question is on your second compound coming, 401. Could you give us the highlights of the differences between 315 and 401?
Yeah, so 401 is a small molecule. 315 is a peptide. So we used a lot of work on understanding what was the important parameters, elements in this host defense peptide that had anti-cancer activity. So we end up with LTX-315 and a peptide composed of 9 amino acids. But in collaboration with the University of Tromsø, we continue to see if we could put more into less. And we succeeded with that. So this is a small chemical modified 1 amino acid. So I think that's quite unique.
Yeah.
So that make that compound a bit different. It is not a peptide that can be degraded. It's more stable. It has some other properties which make them more ideal for deep-seated tumors like liver cancer.
Very interesting. And we have time for one last question. Could you let us know your financial position right now?
Yes. Yeah. So what we reported in the Q1 report very recently is that we have 140 million Norwegian krone in cash. So our cash will last into 2024. Of course, the management and the board is continuously evaluating the position and whether we should add some studies or not, but this is a continuous. But at the moment, we are not discussing any capital raise, so we are in good shape at the moment.
Great. Øystein, thank you very much for the presentation and for being here with us today.
Thank you for inviting me.
Welcome back, everyone. Very happy to have Neola with us here now, and we also have the CEO, Hanna Sjöström, here to present the company for us. I'll let you go ahead straight away, Hanna, and I'll come back later for a short Q&A.
Thank you so much, Eric, and hi, everyone. Great to be here. You can go to the next slide, and we'll start this presentation about Neola Medical. My name is Hanna Sjöström, and I'm the CEO. And today, I'd like to tell you about our vision to revolutionize the care of preterm-born infants with our medical technology innovation that has an extraordinary potential to really upgrade the current standard of care for these very fragile patients by monitoring their lungs continuously and directly warn when complications arise to help save more preterm-born infants, but also save them to a healthier life without severe disabilities. So if we go to the next slide, Neola Medical is a late-stage development company, and we have developed the world's first medical device for non-invasive continuous monitoring of lungs of preterm-born infants, Neola, Neonatal Lung Analyzer. And with this medical device, we are addressing a growing clinical need since we see an increase in infants born preterm. And we have also sold a research device of Neola to an EU hospital, generating real-world evidence for us. Since almost 2 years, we're in the regulatory process to receive an FDA grant, and the same for Europe. And we're now also in the preparation for our US launch. And you'll find us listed at Nasdaq First North Growth Market in Stockholm under the ticker Neola. So if we go to the next slide, why is Neola Medical such an interesting investment case? Well, we have a very large and growing addressable market. So the neonatal intensive care respiratory device market is expected to reach $3 billion in 2030. We also have a great clinical need and demand. So the clinical benefits of Neola device greatly exceeds those of current monitoring methods. We have an attractive business model. It's a classic razor blade model with 1 Neola per bed per baby, and also disposable probe sets on the baby's chest that are changed daily to ensure recurrent revenues. Our technology is proven. Neola has demonstrated positive study results in lung measurements of newborn infants in several studies. And we also have a highly experienced team. So our board of directors, management, and team have a vast experience in medical technology at this advanced level, and also commercialization of innovation. So if we go to the next slide, with our medical device, we're entering a growing and non-cyclical market underpinned by favorable trends and very distinct market drivers. So the global market for neonatal respiratory devices was valued at $1.7 billion in 2022, and is now expected to grow at CAGR of 5.8% to reach $3 billion by 2030. And in terms of the market drivers, there's a very clear need for new medical device in the neonatal care, and it's driven by a number of factors. First, we have a growing number of patients as there is an increase in preterm birth worldwide, leading to more children developing RDS, so respiratory distress syndrome, and other lung complications that needs continuous lung function monitoring. We also have a potential opportunity to really generate high margins globally, but especially in the US, where the pricing landscape is considered to be very favorable. Moreover, there's a rising need and a rising awareness of the importance of newborn disease prevention, leading to an increase in demand for neonatal intensive care respiratory product and services. And since the development of medical device for children is on average 5 to 10 years behind, similar for adults, the US Food and Drug Administration, the FDA, has introduced several incentives and guidance in order to encourage more device development within this field for pediatrics. And that's something that we can clearly see in our meetings with the FDA as well. So there's a real push for medical device towards children to come to market. And if we go to the next slide, we look at the market opportunity for us, and it's really huge. There's a great potential addressable market worldwide because we have that growth in preterm birth, but also an increase in neonatal care clinics around the world. There is a very large medical need for better neonatal care, which also translates into a very high demand for technological innovation. But if we look at the US specifically, that is a very attractive market opportunity for us, especially due to the particular high rate of preterm birth, which is well above 10% of all babies in the US that are born preterm. And they also have expanded their neonatal intensive care, leading to now more than 22,000 beds in neonatal intensive care units, of which 50 to 70% is relevant for our medical device, Neola, according to our very extensive market research with US neonatologists and hospital directors in the US that we performed during 2022. And if we go to next slide, we see the reason why it's so important to have more lung function monitoring in today's care. And it's because 1 in 10 babies are born too soon and may depend on neonatal intensive care for their survival. We have never had this amount of babies born preterm before. It's a huge global problem. And the fact is that preterm birth is the most common cause of death for infants under the age of 5 after pneumonia. And respiratory failure is the leading cause of death in preterm born babies. And it's because their lungs are very underdeveloped. And if we look at the next slide, we can see that today, for the ones who makes it, cognitive disabilities, sight impairment, and chronic breathing disorder is unfortunately the future for many of these children. So given this huge medical need to monitor the lungs specifically and the respiratory problems that these babies have, you might think that there's a lot of lung monitoring in today's neonatal intensive care. But unfortunately, that's not at all the case. So if we look at the next slide, we can see that that's the reason why we have developed Neola, Neonatal Lung Analyzer, a medical device offering non-invasive, continuous monitoring of the lungs with an instant detection of pulmonary complications. And this is to let us save more children, but also let them live a healthy life without severe disabilities. And we're now ready with the product development. And what you see here on the screen, and looking more in detail on the next slide, you see here in real time on the system screen the free oxygen gas concentration inside of the baby's lung, as well as the relative lung volume, both for the right lung and the left lung separately. And the aim here is to instantly detect and provide objective decision support for the doctors for all the relevant clinical parameters, such as changes in lung aeration or lung function development. So here the doctor can scroll back in time to see important things like, is this baby getting more or less stable? So if it's getting more stable, maybe I can pull him or her off mechanical ventilation support earlier, and then save them from all the damages that they today get by lying too long with mechanical ventilation. But also, of course, here the doctors can see instant changes, such as a femtothorax or an atelectasis. Very important to find early on in terms of saving the babies, but also so that they are not lying too long with their complications and then getting all these cognitive disabilities that you have seen. So if we look at next slide, you see here the current standard of care that is in need of new modern technology, especially that offers a continuous monitoring and not only the snapshots of the lung status that is given in today's care. Because preterm born infants are very vulnerable as their lungs are not yet developed, and current competing monitoring methods are clearly insufficient. So what you see here on the screen is today's care, which is a very manual care. You do some chest X-ray, blood samples, also some pulse oximetry to get an overview of the baby's condition. And in some cases, you have lung ultrasound or electrical impedance tomography as well. However, these methods only give that snapshot of the lung status and are very slow to show results. But also all the handling of the baby is very disturbing for the babies and also potentially harmful, especially in terms of the chest x-rays that are done daily today, but also the blood sampling, which is causing a high risk for the baby in terms of blood loss and infections. So with our new monitoring methods, we offer continuous monitoring of the lungs. We're very fast to show results, so below seconds. We're also very safe for the infant's very vulnerable skin, and we're non-invasive, so not breaking a skin barrier, which is exactly what the neonatologists are looking for. So if we go to next slide, we see that what I'm presenting here today is a new precision cure with potential to really upgrade today's standard of care for preterm-born infants. And we believe Neola will be able to contribute to faster detection of pulmonary complications, preventing further major morbidity, so to save more infants, but also a decreased number of days in very expensive neonatal intensive care, which is something especially the US hospitals are very interested in. And then also decreased time for visual observation by the nurses. So today it's a very personal intense care. So for the not so sick babies, you have one nurse per 2 babies looking at the babies in the US NICU today. And for the very sick babies, you have one nurse per baby looking at the baby, trying to see what's going on. So that is the way that we're definitely trying to show in our coming clinical studies that we're also going to decrease the need for that visual observation by the nurses, which is a true cost saver for the US hospitals. So if we look at next slide, so how does this technology work? Well, it's a very innovative and by us highly patented technology that's called biomedical GASMAS. So GASMAS stands for gas and scattering media absorption spectroscopy. So this is a true revolutionary and unique method for measuring volume and composition of gases inside of the body, and is grounded in tunable diode laser spectroscopy. And it was originally developed by a researcher at the Department of Atomic Physics at Lund University here in Sweden. And GASMAS relies on how light is transmitted and is absorbed by the molecules in the body tissue. And in our medical device Neola, the optics provide a very low power near infrared light. So the measurement is completely eye safe, skin safe, and does not produce any heating, no matter the color of the baby's skin. So that's also something that is very important for neonatologists. And the very small and lightweight emitter and detector probes are positioned on the baby's chest, and light is transmitted through the chest cavity and scatters around the tissue. And most of the light is absorbed by the solid and liquid compounds, but a very small portion also passes through the cavities and is absorbed by water vapor and oxygen gas molecules in the lung. And using the data of how light has been absorbed by the gas molecules, we can measure lung volume and oxygen gas concentration in real time, all directly from within the lung. And if we look at next slide, we see here that Neola is also designed in very close collaboration with neonatologists and nurses that ensures that the device fits into the current workflow of the neonatal intensive care unit, both in the US and in Europe. So reaching a commercial market, we have that razor blade business model to secure recurrent revenues. This means that we're selling the Neola device as one unit, and the probe sets that are developed and patented by us are also disposables and are changed daily on the baby. We're also fitting into the existing reimbursement amount at the NICUs in the US, and recently finalized the US pricing study that provided us further insights into the willingness to pay. So if we look at next slide, one way to introduce a really modern ways of working in the care is shown here by the interface of Neola that actually then helps the healthcare professional to guide them through a perfect probe positioning. In this way, we are ensuring that we have a perfect probe positioning on the baby for monitoring, and it's also very simple to use for the nurses. So we're coming into the clinic with a very modern and self-guiding, intuitive approach, as you see here. So there's no way that you can actually monitor the baby without a clear signal and that the screen shows the neonatologist that this monitoring is now live and it's working. So that's one example of how we are bringing very modern technology into the intensive care. And if we look at next slide, we can see that this has been a journey, but I would say for the very advanced technology that we have developed, it's been a quite short one. And it's because the great interest that we see from neonatologists around the world. We started in 2012 with the first gaps of absorption measurements on one infant, followed by 2 more studies in 2013. In total, 32 infants were then measured, and the results then showed feasibility of measuring lungs of infants. And then in 2018, we did a preclinical study that also shows the technology's ability to measure oxygen gas concentration in the lungs and detect complications, which is, of course, also super important. And then we developed a clinical adopted device at the end of 2018, and a clinical study on 12 infants then performed here in Lund showed clinical acceptance for the measurements in general, but especially then also the designs of the probes, which is one way that it's really important to have perfect probes that the nurses and the doctors accept in order to get that clinical acceptance. So we got that. And then we started the product development according to MDR and FDA guidelines in 2020. And during 2021 and 2022, we had a very large investigator-initiated study performed at Infant Centre at University Hospital of Cork in Ireland. And we're now planning further studies this year and beginning of 2024 to show usability, safety, and performance with Neola in an observatory study that we're going to do in the US before we launch. So if we go to next slide, there is also a great clinical support for Neola, and key opinion leaders have said that the technology is both, it's interesting, it's promising, it's super useful, and it has been emphasized that if we can now show the expected results with further data that we're doing this year, Neola could be at every clinic within a 5 to 10 year period. So going to next slide. The technology, as you now understood, is really groundbreaking, and also key opinion leader Professor Dempsey and his team at Infant Centre at University Hospital in Cork in Ireland are also convinced that Neola has the potential to significantly alter the way we monitor babies in neonatal intensive care today, and then especially preterms on mechanical ventilation, not only to monitor them more closely in real time, but also to reduce X-ray exposure, which is very important, and then also limit blood tests that are being performed in today's care. So as a proof point of the great interest that we see from key opinion leaders, we have already now, before CE marked, sold a research version of Neola to University College Cork for approximately $64,000. And the research team has used the device in a very large clinical study on 100 newborn infants. The results of the study were presented in the US in last year, in 2022, and the results from the study confirms that the technology works well for measurements of newborns' lungs, which also strengthens our perception of Neola's future position in neonatal intensive care. Another study that this research team also performed was presented just a few weeks ago on the largest neonatal intensive care congress in Washington, and further publications are expected this year, which we are, of course, following very closely. So if we look at the next slide, we are following a very distinct path to market launch. Milestones are important to us, and here you can see them more in detail. We keep to our established plan and we reach our milestones accordingly. And now we are entering a very interesting phase. It's a commercially focused phase where we mainly work with pre-launch market activities, technical verification and clinical validation studies, and the regulatory process that is ongoing with authorities in Europe and the US. If we go to the next slide here, you can see our commercialization strategy and plan. And our commercialization plan is divided into 3 phases, pre-launch, launch, and scale. We're currently in the pre-launch phase where we're focusing on gathering scientific support and gain publications from key opinion leaders. We're also visiting neonatal intensive care clinics and conferences, and we're very active in digital forums for neonatologists interested in the latest innovations. We're also in the regulatory processes for the FDA approval and CE mark, preparing for market launch in the US and Europe with a US first focus. And we're expecting the launch phase to start in 2024, where we're targeting key teaching hospitals in the US with very large NICU departments to get those early adopters. And following this, we also see a phase with scaling, mainly within US and Europe. And if we go to the next slide, we have a long-term strategy to build a very strong patent portfolio in order to enable consistent value growth. So our patent portfolio is continuously strengthened, and we have 6 patent families covering all major assets, both from the medical device side and technical and medical patents. And we own all relevant IP related to the device and disposables. Now, what is also important from an IP perspective is the future applications. And we just received a patent granted in the US for a method that let us monitor larger lungs, such as children and adults. And this is an important future applications, and we definitely see a growth potential, especially in the care of older children in intensive care and during surgery. So therefore, I was really happy to be able to announce that in March, we were granted funds from Vinnova for 3 million SEK for a research project together with Lund University and Lund University Hospital, where we, together with intensive care doctors and technical researcher, are now exploring the clinical potential as well as the business potential for monitoring children, so expanding our patient group. And if we get to the next slide, we do this journey with a highly experienced team that we have also expanded this last year, and also with a strong board of directors with experience both from the field and from commercialization within medtech innovation. So finally, next slide, please, here. So why invest in Neola Medical? Well, to summarize, we have a very large and growing addressable market. As I'm sure you understood by this presentation, also a great clinical need and demand, very attractive business model, proven technology, and a highly experienced team. So with that, and next slide here, I want to thank you for listening, and do join our journey to improve the care of preterm-born infants. Thank you so much.
Thank you very much for the presentation, Anna. We have about 2 minutes left, so I will try to squeeze in 2 questions at least. So first off, I mean, what sort of potential do you see for reimbursement coverage in the US? Will this be a situation where wards will have these devices and sort of attract patients by having them, or do you think there's a scenario where they could get full reimbursement coverage?
So what we see today from our reimbursement analysis is that we will be covered by the overall amount that the NICUs get for reimbursement of the children. And we're fitting well into that model already, so we don't need a special reimbursement for the Neola device to be used. Important then with that model for us is to really get key opinion leader support, because what we understand from today's care, especially in the US, is that what the neonatologist wants for the baby that is potentially rescuing lives, they will get. So it's really important to have that full buy-in from the strong neonatologist within their department. to get it. But in terms of what we also hear from neonatologists, it will be a marketing factor for them as well to be able to attract the parents who are well understanding of the situation for preterms and who wants to be in the university hospitals where they have the latest technology.
Okay. And then a strong set of data is usually at least very beneficial for breaking ground in the US. How much more data do you think you need to generate? Do you need to run any large-scale studies in order to market this successfully?
So in the first step, we'll do an observatory study with preterms in the US. And for that study, we will have what's needed for an FDA grant. We evaluate today that it is sufficient in order to take the product to market as well. And then we'll follow up with larger multi-center studies in order to add more clinical claims and also to show more improved clinical outcomes after we have come to market.
Perfect. All right, that was all the time we had today. Thank you very much, Hanna, for being here with us. We'll now take a short pause and then return with Alcavello. Thank you very much.
Hey everybody, thanks for joining us again here. And now we got one of my favorite allergy companies coming up. It's ALK at Bello, and with us today we have Per Plotnikov, VP Corporate Communications and IR, and a good old friend. So Per, take it away and tell us why ALK is such a great company that we all think it is.
Thank you very much, Morten, and thank you all for listening in and for the invitation to present today at your conference. Let me just start by setting the scene. So if you please turn to slide number 2. And for those of you who don't know ALK, let me just give you a brief introduction. So ALK is the global leader in a therapy area called allergy immunotherapy. We have more than 100 years of experience in allergy diagnosis and treatment. We are the market leader with more than 40% market share, which we continue to increase. We currently treat more than 2 million patients with our products, and our objectives is to help ever more patients to take control of their allergy and improve their quality of life. Our products work by re-educating the immune system, alleviating symptoms, and offering long-term protections. Our core products, they are the sublingual allergy tablets that cover the world's most important respiratory allergies, and which have the potential to become mainstream treatment for poorly managed allergies. And they currently make up approximately 50% of our revenue. Over the past 20 years, we have made significant investments in these products so that the portfolio today is the world's best documented product portfolio in the history of allergy immunotherapy. And they are strongly protected by very unique biological manufacturing processes, meaning that essentially that there is no patent expiration issues associated with our portfolio. That's of course exceptionally important also from equity story point of view. ALK is well positioned to deliver on our goal of achieving 10% top-line growth average annually with increasing profitability. And to do this, to achieve this, we are increasing our lead in respiratory allergies by gaining market share, but also by expanding the overall market with tablets as a key driver. We also expect growth from the non-tablet portfolio. A major focus of ours is market expansion into new geographies, into new segments, for instance, the pediatric space, which is a very significant expansion opportunity for ALK and which is very soon to materialize. As part of our strategy, we're also building new businesses with the potential to accelerate our long-term growth. For example, we have just entered into food allergy, starting with a peanut allergy immunotherapy program. So the ultimate goal of ALK is to become the allergy company, a global allergy player that helps millions of people take control of their conditions and of their lives. Please turn to slide number 2. Allergy immunotherapy today is a relatively niche treatment. We treat roughly 5 million patients in the industry. ALK has a set 40% market share. So the 5 million patients, that's actually only 1% of the people suffering from allergies. We should be treating 10%. We should be treating 50 million people. And our tablets, they are designed to expand from the 1% into 2%, into 3%, etc. Over time. When we look at the allergic patient journey, a lot of people, they suffer unnecessarily, and they suffer for many, many years. It's a very long and convoluted patient journey. And our entire strategy is very much focused on bringing more structure to the patients, giving them better opportunities and better access for proper treatment of their condition. Please turn to slide number 4. Our strategy is essentially summarized in this slide. So the strategy is all about expanding our lead in the respiratory field. It's about educating and empowering more people to seek help for their condition. And of course, now it's also about expanding into food and also expanding our anaphylaxis offering. Anaphylaxis being an acute, severe allergic reaction to, for instance, bees or wasp stings or peanut, etc. Our strategy has 4 pillars. Geographical expansion, North America is key. I could also have listed China here. China, we are currently in regulatory review with one of our key products. We expect approval next year. That's also a major growth opportunity for ALK. We continue to invest in the US. US is a little bit more tricky in allergy immunotherapy, because here we work with a system, with a group of doctors that have a very strong financial incentive to protect what they have been doing for the past many centuries. So for us, the US is very much about building new channels. For instance, right now we're focusing a lot on moving into the treatment of younger patients, and also focusing more and more on doctors that are like outside the traditional allergy specialist segment. So moving more into the ped space, and focusing more on new treatment channels. The second pillar of our strategy is a focus on our tablet portfolio, making sure that we complete the development of this portfolio, especially for pediatric use. This year, we are finalizing 2 very important phase 3 studies. One that very soon will read out data. That's the pediatric study on house dust mite, phase 3 study in 1,500 patients covering Europe and North America. We expect results here mid-23. The second one is with our tree tablet called Itulazax. That one is expected to read out top-line results in Q4 of this year. So very eventful year. We are, of course, also focusing a lot on driving the commercial momentum with tablets. We've had a little bit of short-term challenges in some of our key markets. We believe they are temporary, and we are convinced that tablets, they remain key to ALK's growth, not just in the short term, but also medium and long term. The third pillar of our strategy is consumer engagement and new horizons. So essentially, a key factor for why allergy immunotherapy is so underpenetrated in the bigger overall market is the lack of awareness of this treatment option. So our focus in our strategy is also to expand awareness, especially via our digital platforms that we have been investing in over the past 5-6 years, with the aim of engaging with people who have allergies, identifying the candidates for immunotherapy, i.e. the ones that have poorly managed allergies, and then help mobilize them towards doctors that are able and willing to help them. And we do this under a brand called the Clarify Universe, and which is essentially a digital universe that informs people about their allergy, help them navigate their allergy, but also help them get access to doctors via Find a Doctor tools, via helping them to book an appointment with the doctor, etc. So it's very much a patient mobilization objective that lies behind these consumer efforts. Under the new horizon, this is our food investments, developing a food allergy solution, building on the efforts that we have been doing over the past 20 years. We are now taking this a step further and are developing a peanut allergy medicine for all the kids that are suffering from severe peanut allergies. We are currently in phase 1. We also this year expect to see the first initial results from this phase 1 study. So in many ways, 23 is a key year for us when it comes to the R&D data. Then on the final and fourth column of our strategy is also making sure that we run a very efficient business. When we launched this strategy back in 2017, we were, as a company, somewhere between a rock and a hard place. Not only were we required to invest quite a lot in our business model, in our R&D, but we were also required to clean up in our legacy business, which was seeing a lot of challenges and problems with quality, FDA challenges on warning letters, injunctions in France, etc. So the consequence back then was to make sure that we clean up our portfolio, we remove all the products that are non-sustainable, and we optimize the ones that will be on our product portfolio. And so far, we have taken out more than 300 product variants across our markets, mainly in Europe. That's equivalent to 10-15% turnover that we have removed in order to have a strong fundament to stand on. The focus right now is, of course, to optimize our manufacturing footprint so that we get the best possible leverage out of the top line growth that we, of course, is core for this equity case. Please turn to the next slide. So that was the big picture, and just zooming a little bit in on the short-term development, and a little bit also on the reasons for the recent setback, also in the share price that we have witnessed. Q1 this year saw actually overall results that was largely as expected. However, our core products, the tablets, they came in 35 million short of what we had expected. And this was mainly related to the fact that we initiated slightly fewer patients in our key markets of Germany and the Nordics. We failed to initiate round about 10,000 new patients. While we still initiated 200,000 patients, we had expected to initiate 210,000 patients here over the 22-23 pollen season. The other markets, North America, Japan, international markets, were actually doing as expected, although some of it was influenced also by phasing of shipments. But the core to this falling short of expectations on tablets was very much linked to Europe. We are convinced that this is a temporary effect and not a structural issue, and we are focusing a lot of our sales and marketing activities now on making sure we get a strong recovery in growth in Europe in the second half of this year. Right now, in Q2, it's the low season for new treatment starts, even though it's the high season for pollen allergies. But when we have pollen in the air, we typically don't initiate new patients on treatment. That's something we do once the pollen season has ended. So right now, all our marketing campaigns are focusing a lot on making sure we have a strong trajectory coming into the second half of this year. While the tablets were a little bit below expectations, and which obviously, as I'm sure you are aware of, caused a very strong reaction in the stock markets, our legacy business, which we abbreviate SCIT, stands for subcutaneous immunotherapy or injection-based immunotherapy, they actually performed quite well with double-digit growth in a mix between volume growth but also improved pricing. And we also saw our operating profits increase in the quarter, despite us continue to invest a lot in our strategic growth initiatives. Please move to slide number 6. So on the next couple of slides, just a little bit of focus on what are we doing in order to restore strong growth in Europe on our core products. So the short-term actions here are very much focused on mobilizing patients. This year, we are so fortunate, if I may say so, which is, of course, perhaps not the right way of setting it up, but we are in a situation with a very strong pollen season. So people suffer badly from their allergies. So our focus is, of course, to mobilize as many patients as possible. So we are running a lot of campaigns on our digital platforms aimed at engaging and mobilizing people to take action on their allergy, to go see a doctor that can help them with a long-term solution for their allergies. We're also focusing a lot of our sales and marketing efforts on mobilizing allergy doctors. So historically, and this goes back many, many years, the clinical practice around AIT has been, so we initiate new patients in Q4 and partly into Q1. That gave us some challenges last year because in Q4 we also had other external influences impacting the patient flow into the clinics. It was impacted, for instance, by a number of respiratory infections that crowded out some of the allergy patients from the clinics, doctors were preoccupied with dealing with these respiratory infections, and which was essentially the cause of us falling short of these 10,000 new treatment initiations. So a key message in our campaign now is to get doctors to start initiating people earlier than usual on AIT. So already start to initiate some patients maybe already in Q3. So that is what we're campaigning heavily right now. We're also focusing on expanding the prescriber basis to increase the capacity so that we can get more and more doctors to actually start treating people with severe allergies. And then finally, we're also working with improving pricing, partly as a response to, of course, inflation, etc. And we're also working on improving market access across the board. Next slide, please. If we take a little bit longer view, I think the tablet outlook remains strong globally. And on this slide, I listed a couple of the medium and long-term growth drivers of tablets. Of course, right now in Europe, if you take that as a starting point, our core tablet markets are Germany and the Nordics. These two markets, they make up the bulk of our tablet sales. So a focus of us in Europe is, of course, also to go beyond these markets. We still want to grow these markets, but there are also plenty of growth opportunities in other of the Central European markets, in UK and in South Europe. Each market has its own characteristics and own challenges that needs to be dealt with, but we are focusing on making sure that they also become meaningful growth contributors going forward. As I said already, we focus a lot on expanding into the pediatric space. If we have positive phase 3 data this year, then we will be able to start launching the tablet portfolio into the ped space from next year and onwards. And that will be a structural important growth driver for ALK, not just medium, but also long term. If you look towards Japan, where our products are already approved for children, we actually see a very strong traction with AIT in young children. 80% of all new patients out there are actually kids. So that's super important. And Japan generally is actually a very important market for us. There's a lot of very positive movement now. Allergy is a big thing in Japan. More than 40% of the population they have allergies, and the government in Japan is very focused on helping out on this situation. So for us, Japan has been actually a very strong success story, and we do also expect it to be going forward. And there's a lot of movement right now in Japan. So Japan is also a market to watch. And then, as also mentioned, a launch in China, hopefully from next year, conditioned upon approval that we are currently in process with in China. And when we look at China, it's very much about house dust mite allergy. It's a very big thing in China. It's very much associated with the urban lifestyle. And we have been expanding and investing in our infrastructure in China over recent years. We already have a legacy product approved in China, an injection product, which is actually growing quite nicely. And we use this as a way to pave the road, so to speak, for the launch of the house dust mite tablet next year. So China is also very promising. We also have a pretty risk measured approach to China. We have stage-gated approach so that we will not invest everything up front with the hope of a return someday. We are investing, we are opening up for the next tranches, only conditioned upon meeting a lot of interim criteria for hospital access, for advocacy, etc., etc. But China looks quite promising. At the same time, we have also continued to invest in US, and even though the US has been the big promise in this equity case for many years, it's still a long case. It's still slow moving, but we continue to invest because the option value in the US is so big. So with this, please move to next slide. So in summary here, the outlook for 23 is unchanged. We changed the product mix a little bit here in connection with Q1. We still expect 7 to 11% growth in local currencies. which is equal to 8 to 12% growth if we just disregard a 1-year temporary increase in some mandatory rebates in our core market of Germany. We are now looking at sales of our legacy products of around high single digit, and we do expect tablet growth 9 to 14%, with the high end of that range reflecting some extraordinary price increases that we believe we are subject to. And what is also important to manage expectation, we do expect revenue growth to pick up from the second half of this year. So don't expect significant changes in the European growth outlook here in the second quarter, again, because we are in the low season for new treatment starts. All what we do now is to make sure that we have a strong trajectory going into the second half of the year. If we look at EBIT, our earnings, we expect earnings of 13 to 15% margin this year. We expect the gross margin to improve. We expect R&D to be around 600. And we do expect sales and marketing costs to increase, but not as much as sales. So we do expect mid-single digit growth in sales and marketing. So we do also expect to see leverage on our earnings from top line growth, essentially. So the capacity cost to revenue ratio is expected to improve still. Please move to the next slide. So this is the final slide, and just summarizing, we work with severe allergies. There is a substantial need in this space. Many people, they suffer unnecessarily in silence because of lack of awareness, lack of access to adequate treatment. That is what we are working to improve. So we're very focused on expanding our AIT leadership and engaging more closely with patients. We are a growth company. We need to sustain high growth. That's our raison d'etre. That's our justification for the long term. But we also need to translate that top line growth into improving profitability, while at the same time investing in new businesses that can safeguard or potentially accelerate the long-term growth of the company. So with those words, that was it for on the introduction on ALK. And Morten, I think now there will be time for a question from the audience or from you. Thank you.
Thank you very much, Per. It will be me. We decided that I would be the bad guy here. You laid out the weakness for Q1 quite nicely, but you also talked about why it should improve. But still, at the core of this was that the traditional growth driver, the tablets, fell short, and it's a bit unclear why. But you still have the skid slit actually growing very nicely. It's a bit of an odd mix here, especially the change in mix between the two business units. Is there any reason why the skid slits would do so well in Q1 versus the tablets?
It's an absolute fair question. So the tablets were impacted by these respiratory infections that essentially meant we fell short of these 10,000 new patient initiations because so many patients were preoccupying the clinics. The reason why injectables, SCIT in particular, was growing was a mix between improved pricing. So we had quite significantly improved pricing in some of our key markets, both Europe and North America. But we also had volume growth. So we had volume growth in China on our injectable, but also in Europe. And in particular, last year, we had some supply challenges in parts of our injectable business. So just as a reminder, our injectable SCIT segment, it covers many allergies. So it's a portfolio of products that lies behind that one. One segment here is the venom space, which is roughly 15-20% of that overall segment. And last year, we had some supply chain challenges here. And this year, we had a strong recovery in this space. We had also significantly improved pricing on these products at the same time as some of our competitors were also challenged with supply issues. So we were benefiting here in a subsegment of that space that is a little bit different to the traditional respiratory space. So it's a mix between volume and pricing.
Okay. One more question for me, because you talk about the improved pricing on skid slits. But you're also hinting at, in your guidance for 2023, that there may be some upside towards the higher end of the guidance range if you are successful in driving prices for some of the tablets. I mean, many of the pharma companies has been able to show some improved pricing over time, especially in light of the inflation. Why shouldn't we expect you guys to also do this?
That's also part of it. But then we also have these extraordinary price negotiations that we are in one of the European markets. There is definitely, and I agree with you, Morten, that there are opportunities under the umbrella of inflation compensation also to improve general pricing in parts of Europe, and we are certainly pursuing that. That's part of the guidance also. But on top of that, we believe we are entitled to some improved pricing on some of our key products because we have had an expanded indication. This is one of our key markets. Now this is uncertain. This extraordinary price improvement, because that's linked to some product specific dynamics. But I would agree with you, and that's also part of our guidance, that overall improved pricing because of inflation, that's those possibilities we certainly pursue, and that's part of it also.
Thank you very much, Per. We are unfortunately running out of time here because there were so many more, but thank you very much for joining us here today and outlining what we feel is a very interesting story here for you guys. And let's see what you got to report to us in August at the Q2 numbers. Per, thank you very much for joining us here today.
Thank you, Morten. It was a pleasure, and please enjoy the rest of the day.
Thank you very much, Per. Talk soon. Bye-bye.
The next company presenting today is Exact Therapeutics. Exact is a biotech company developing a new ultrasound technology for therapeutic enhancement. And with us, we have today here the CEO of the company, Per Walday. Per, the stage is yours.
Thank you so much, and thanks for the invitation to present Exact at the Life Science Summit. If you can move to the next slide, please just pay attention to this important notice and disclaimer. And then on the next slide, you will see some short, brief company facts. Company is a spin-out of GE Healthcare, and it has a long history of R&D. It's been a total of 15 years of R&D and more than $35 million invested into developing this very unique platform technology that ACT is. ACT stands for Acoustic Cluster Therapy and can provide targeted drug delivery, and that's what we're doing in Exact Therapeutics. It's broadly patented in major markets, including the product composition of matter, the product PS101, and we are now in early clinical phase. As you can see on the left-hand side, it's primarily strong Norwegian family offices, long-term, sustainable, supportive investor base, also some institutional and the founders, and a small retail share on the shareholder list. We were listed on Euronext Growth in July 2020, and we're now looking to expand to a more global investor base. We have a strong industrial collaboration still with GE Healthcare, both with regard to supply of PS101 and also technology development with regard to the ultrasound, the technology that we're using for this drug delivery technology. We also have strong academic collaborations with several expert sites with deep domain knowledge. We have TGen, Translational Genomics Research Institute in Phoenix, a strong collaboration with the key opinion leader within pancreas cancer, Dan von Hoff. We have Institute of Cancer Research at Royal Marsden, London, and also Norwegian University of Technology in Trondheim, where we do a lot of preclinical experiments. If you go to the next slide, you will see just that brief overlook over the leadership. We have a really strong leadership, global leadership team with a lot of industry experience. The chair of the board of directors is Anders Wold since a year. He was part of starting up WingMed and building WingMed, and when it was acquired by GE Healthcare, he over time also became the CEO and president of GE Healthcare Clinical Care Solutions and Ultrasound, a total $6 billion business in GE Healthcare, and he had that position until very recently, a little bit more than a year ago. Masha Strömme has been with the company for a long time. We also have Bill Castell, Sir Bill Castell, who is the former CEO of GE Healthcare and also has been the chairman of the Wellcome Trust. So a lot of network and deep industry expertise in the board of directors. The executive management has a long industry experience in bringing products from research all the way into the commercial phase. If we go to the next slide, I'll just briefly touch on something that is happening in healthcare these days, and you all know this. Precision medicine is on the rise, and oncology is the vanguard of precision medicine. We know that immunotherapy and pharmacogenomics, if not already, they are trending to dominate the oncology market at some time point in the near future. With precision medicine, you need often very targeted medicines, which are biologics, large molecules, sometimes fragile, need to be packed into vehicles, which can, in some instances, really produce challenging tissue distribution to get these out of the blood and into the tissue or the pathology where you want them to work. It goes across a number of different areas like immunotherapy, nucleic acid therapy, gene therapies, and packaging systems of liposomes and nanotherapy, etc. And there is a need at the critical junction now for targeted precision medicine delivery methods. And ultrasound is something that is on the rise. Moving to the next slide, you will see that, if you can move to the next slide, please. you will see that it's really an emerging treatment modality. Some announcements that was done in the beginning of the year about different companies that are showing progress, and also that we can see that the number of the pharma VCs and other are moving into this space. At the bottom on the right-hand side, you can see the increase in patenting around ultrasound microbubbles for therapeutics. It's been a fantastic rise in the number of patent publications per year the last decade. So it's really gaining momentum now as a new promising treatment and drug delivery modality. And most companies are using already existing small microbubbles to enhance and use this technology in vivo for medical purposes. What we have is a proprietary and completely unique non-invasive platform technology that is specifically designed to enhance that clinical benefit, to enhance the drug delivery. So let's have a look at the technology. On the next slide, it's just the intro slides. You can move one more to the technology bit. PS101, that's our core product. It's a highly engineered, unique prodrug technology. It consists of a gas bubble, the PFB, the red bubble here, perfluorobutane filled gas with a phospholipid shell, which is negatively charged. In addition to this, we have another perfluorocarbon oil emulsion with a positively charged shell, and these attract each other. So in this formulation, you have small clusters of gas bubbles plus these oil droplets. So that's a prodrug that we can inject and that flows freely in the blood. And when you apply ultrasound on an area after injecting this, you will activate these small clusters. And during activation, the oil emulsion, the oil droplet, will go through a phase shift and evaporate into the bubble and create a larger bubble that is trapped in the capillary system. So it converts the clusters to large act bubbles, and they are temporarily 5 to 10 minutes trapped in the capillary. And then you can start manipulating for these 5 to 10 minutes. You can manipulate these larger bubbles and sort of prepare the tissue for targeted drug delivery, open up the endothelial cells that the capillaries are consisting of, and also breaking up collagen bonds and opening up the fluidity of the microenvironment around cells, and thereby making it easy for medicines to pass from the blood into the tissue. And then the gas in these act bubbles will dissolve into blood over time, and being released from the capillaries, and the perfluorobutane and PFMCP will disappear from the body within hours. On the next slide, it's a bit more details on what's happening. We have these clusters passing through in the capillaries in the blood, and then with normal low energy diagnostic ultrasound that is used today for a number of different purposes in every hospital in the world, you can apply that onto a specific target non-invasively through a probe, and then by this in milliseconds activate this to become a large bubble that is trapped in the capillary. And then changing the frequency to a lower 500 kilohertz frequency, which is closer to the resonance frequency of these larger bubbles, you can start to make them oscillate. And when they oscillate, they will break down physical barriers that are hindering medicines to pass from the blood into the tissue. And this is like a preparation of the tissue, because the effect you don't need to have the drug at the same time as the bubble. You can have the bubble and do the preparation first of the tissue and then give the drug afterwards, and you will see that it increases the delivery of the drug into the tissue where you have done this preparation. On the next slide, you can see the advantage that we have with the ACT bubble. So what the others are trying to do is using a diagnostic microbubble that passes through free-flowing a few milliseconds in the capillary before it disappears. And to try to get some effect out of this, it is not in direct contact with the endothelial cells. It's confined to the vascular space, while the ACT bubble is 1,000 times bigger in volume and in direct contact with the endothelial wall, with the cells that constitute the capillary. And then when you oscillate this, you can stretch it from each other so that it opens up and become more available for drug. And this has also been modeled in different models, and we know that this produces radial tissue displacement, and it displaces tissue and creates shear forces that goes beyond, far beyond just where the capillary is, and across probably several capillary levels, so that you prepare the tissue in general where you make this happen. So with this technology, what kind of results have we achieved? So if we move then to the preclinical part of the deck, next slide is the entry slide for the preclinical. So if you move one more, this is the result from a study that has been done in pancreatic ductal adenocarcinoma, a PDAC model where you use patient-derived material in a xenograft mouse model. And this has been done by TGen in Phoenix by Professor von Hofs, one of the most known global key opinion leaders in pancreatic cancer, that was behind the studies for approval of both of these drugs that are tested here, nab-paclitaxel, which is also called Abraxane, plus gemcitabine, and liposomal irinotecan, which is also called Onivyde as a brand name. These drugs are standard of care in pancreatic cancer, and what you see on the right-hand side are the curves of the growth of this tumor. And you can see on the black that these tumors grow quite quickly in the mice. When you give nab-paclitaxel and gemcitabine or Onivyde as a standard of care, you can see that you can put a brake on that. It stops the growth. It doesn't really shrink on average the tumors very much, but you can stop the growth for a period before it starts to grow again. When we apply ACT on top of the standard of care to enhance the delivery of the standard of care into the tumor, we get a real significant effect with 50% of the animals, the tumor disappearing completely. We have tried to do this only with the PFB microbubble, with the gas bubble without the oil component in the same study. And it turns out that that doesn't have any effect at all when we use the same ultrasound procedure that we use for ACT. In addition, I would like also to mention that this is a patient-derived xenograft mouse model, and the reason for using this is because the morphology, which is very difficult to penetrate in pancreatic cancer in man, more resembles the man version than many of the cell line pancreatic models. So Dan von Hoff have done this study, seen these results, says that this is some of the best results he's ever seen in this model, and he's very eager to see this moving into the clinic and to get some patient benefit out of this technology. We have also looked at blood-brain barrier and CNS. If you go to the next slide, you will see that a mice model has been used to see whether we can break the blood-brain barrier, which is a kind of a barrier between the blood and the brain to protect the brain from any harmful substances that can be in the blood. So it's really a protective barrier. But it also means that most drugs, only 2% of small molecules and almost no biologic drugs can enter into the brain because they can't pass the blood-brain barrier. So a way of destructing, or not permanently but temporary, open up the blood-brain barrier, especially if you can do it targeted for specific areas of the brain, would be quite beneficial with regard to having new drugs, being able to treat diseases, neurological diseases or tumors in the brain. And this is what we've done here. We've used the technology to open up the blood-brain barrier in a specific region that you can see the ACT-treated area on the right-hand side, and in MR imaging to see the increase in gadolinium. And when we gave micelles that are fluorescent, you can see the increase in the intensity. And taking out these brains afterwards, looking through whether there's any pathology, we couldn't find any hemorrhage or any pathological consequences of this. It's a temporary and safe way of targeting opening of the blood-brain barrier in the CNS. So that's another application. If you go to the next slide, it just summarizes that we have a lot of strong preclinical evidence across different models and across different drugs. We've done proof of concept for opening the blood-brain barrier. And on the right-hand side of this line, mice cancer models, pancreas, breast, colon, prostate, and combining it with different cancer drugs, 6 different cancer drugs included, and ACT strongly enhance the efficacy of all. And we're talking about increased and complete disappearance of cancer when we apply this technology together with standard of care or other drugs. In addition to that, preclinically, we've done extensive safety studies performed in many animal models, and we haven't seen any significant adverse effect of the ACT treatment. So it seems to be very safe. One of the concerns when you have a technology where you create larger bubbles in the blood would be if you have any ischemic events due to this. And we haven't seen anything of that in the studies that we've done, also with relatively high concentrations and activating in the left ventricle of the heart to ensure that we activate as much as possible and that it spreads to all different organs. So with all of these results, we really have a platform technology. And if we go to the next slide, I just want to have one slide following this on the platform development strategy. We can use this both, as we've seen, to enhance standard of care medications, but maybe more important, novel therapeutics in precision medicine to make sure that they are delivered into the tissues and organs where you want them to work. Our platform strategy here is to start with oncology. That's where we have our focus. We have really strong preclinical data, and we are in the clinic here. So what we want to do is to demonstrate clinical proof of principle with standard of care therapeutics in oncology. And then in parallel with that, we are working on platform expansion through preclinical programs, preclinical studies in different areas, and with third-party collaborations. We are also looking in oncology with regard to immunotherapy. We have a research program that has been supported by a $1.6 million grant by the Norwegian Research Council, where we work together with TGen, with ICR, and with NTNU to look at the immunological effects of ACT by itself and how we can enhance the effect of immunotherapy, which is a fantastic methodology for treatment of cancer. But unfortunately, very many patients do not respond to this. And one of the reasons might be that you don't get sufficient material into the tumor. CNS, as we've shown, we've seen BBB escape, blood-brain barrier escape demonstrated already. So we're looking to collaborate with ultrasound hardware provider because you need specific ultrasound hardware to go through the skull and work in the brain. And then, of course, gene and cell therapy, we're looking to get an established collaboration with biopharma companies in that area to drive that part of the platform utilization. Now, moving to the next slide, I would like to spend some time on where we are in the clinic. Before I move into the study itself, just to tell you a little bit of the indication where we currently are testing this. And this is with liver mets, patients with liver metastatic disease. That will be a top future killer in cancer. GI tract cancers, from the gastrointestinal tract, they are overall increasing in the US, and they are affecting younger people, especially colorectal. So a large increase in pancreatic and liver and bile duct, and colorectal younger people. And it's going to be 3 of the 4 top causes of cancer deaths in the US. A large proportion of these patients are expected to die from uncontrolled tumor growth in the liver. So there's a high need of better treatments for liver metastatic disease. Next slide, please. So this is then a phase 1 study with the ACT technology in patients with more than one liver metastatic liver met from colon cancer. And of course, it's phase 1 primarily. It's done at Royal Marsden, the Institute of Cancer Research. But we also have designed this so we can evaluate efficacy. If you go to the next slide, it just quickly shows that we are applying this together with standard of care. So we don't change the standard of care. We just add our PS101 and an ultrasound probe targeting the liver lesion that we want to sonicate. On the next slide, you can show the design, the concept. One lesion is sonicated. We have more lesions than one. Then we compare at baseline and follow up. How much do these shrink when we apply standard of care with and without ACT? So we have an intra-patient control. And what we want to see is something on the right-hand side, that we have more shrinkage of sonicated than you have of control lesions. We have, on the next slide, released a couple of patients so far. We will come with the full results. And we have some very encouraging early signs. Similar responses to what you see here in patient 3 and 4. We've also shown patient 2 previously at an ESMO conference, have been seen in most of these hard to treat patients. This is often 6th or 7th line of treatment, and we haven't seen any serious adverse events. So based on this, ACT treatment does not add to the safety or tolerability. On the next slide, where we are currently and where we're going. So dose escalation, we're about to complete that. We'll have results in the autumn. And then we do an expansion where we randomize patients to 2 different dose levels to determine which dose to take forward, and also to get more solid efficacy data and safety data. And then there is a part 2 population expansion afterwards that we will come back to when we have done phase 1 finished. So lastly, on the last slide, next slide, please. With unique platform technology, with really strong preclinical data, we are in an emerging field right now, and we are in the first mover position. There's really straight commercial adoption because this probe, this technology can be applied to consoles that are already out there in the hospitals. It's ultrasound is ubiquitous, it's everywhere, and it can be built on the installed base. And we have a really competent, experienced leadership with strong, deep industry experience within ultrasound and within medical treatments. So with this, I think I have used a bit more than my time, but not much. And thank you for your attention.
Thank you very much, Per, for your presentation. Very nice. Now we have a few minutes for questions. I guess that your technology, your ACD technology, will be run by an operator. And I was wondering if you could give us some color on how do you see this happening in the future in terms of potential training for these people and like implementation in hospitals?
So ultrasound is used today in hospitals by radiographers or radiologists in Europe, primarily, sonographers primarily in the US, which are really professional people in doing this. And it is normal diagnostic ultrasound. So it is only the way they today locate and find tumors when they do radiofrequency ablation or other more invasive technologies. This can be done also with this technology in the same manner. But it's just to sort of push a button to manipulate these bubbles to get more drug delivery. So it's a quite easy implementation, and the training is not very extensive to use this. It has the same imaging quality as normal ultrasound. So in the US, it primarily be sonographers, and in Europe, radiologists using this, together with the oncologist, of course, for oncology, or neurologist for neurology applications.
Interesting. And how much variability can you expect from interoperator, let's say?
It shouldn't be too much because it's relatively easy to cover, and we have built navigation module together with GE Healthcare on this. And you can build that navigation module so you can actually see how much treatment you get in different areas of the tumor. You can have it going from red to green and be completely green when you've covered everything and the treatment is finished. So we're working on ways to make this as user-friendly and easy as possible, so that sonographers also in the US can use this technology.
Great. And one question in terms of dosing. What can you say there? I mean, is it possible, for example, let's say a chemotherapy or an immunotherapy is administered once a week? Could you be able to apply your technology also once a week, or you're limited in terms of not disrupting the vascular wall that much or that often?
No, so we're not limited to that. But what we're doing today is just following the standard of care procedures for the FOLFIRI that we are using in the phase 1 study. So for each of the cyclic treatment they get every second week, we also apply ultrasound. That's not a problem at all with this technology.
Oh, great. And I see that your pipeline is mostly focused on oncology. Do you see opportunities beyond oncology, let's say local infection?
Local infection is absolutely a possibility to enhance the delivery into biofilms or hard-to-treat infections of antibiotics. There are a number of applications because, I mean, within neurology, I've already mentioned opening up the blood-brain barrier targeted to different parts for Parkinson's disease, Huntington's chorea, ALS, it could be Alzheimer's, a number of different potential applications that can be done. And also any other organ where you can reach with ultrasound, which is so more or less the whole body, where you want to deliver hard to deliver drugs. Could be nucleic acid therapeutics, gene therapies of different kinds, and so on. So it's only really the application. It doesn't penetrate bone very well, so deliver things into bone if it's difficult. It doesn't penetrate gas very well, so lung is not the preferred organ. But apart from that, it's possible to apply mostly everywhere.
Great. And one last question. Could you remind us your cash position and your cash runway?
So our cash position right now is around 5 million euro. We have a runway in towards the end of first quarter next year.
Okay, great. Per, thank you very much for being with us today and for your presentation.
Thank you. Thanks for the opportunity to present. Thank you very much. Bye for now.
Hello and welcome back, everyone. Very happy to have Photocure with us now for what I believe is the last presentation of the day. So great to end it with a high note. And we have the CEO, Dan Schneider, calling in from the US to present the company. So good morning, Dan. I'll let you go ahead with the presentation right away, and I'll come back later for the Q&A.
Okay. Fantastic. Thank you. And thank everyone for hanging on today. Apologize, I can't be there in person. I've got multiple trips, I think 3 to Scandinavia in the next 3 weeks, and I couldn't pull off a 4th this week. But I'm happy that ABG asked us to present virtually. So let's get started. I'm Dan. I'll be presenting the presentation today. On the line with me also is Eric Dahl, our CFO, and David Moskowitz, Vice President of Investor Relations, for the Q&A portion along with me. Next slide. So today, just remind everyone, disclaimers are in place. Let's go to slide 3. So just a little background on Photocure for those who aren't familiar with the company. We are a global company headquartered in Oslo, Norway. We're traded on the Oslo Stock Exchange, have been around since 1997, launched the current product, which we'll get into momentarily, in and around the 2005-2006 timeframe in Scandinavia, or the Nordics, and then Europe a couple of years after that in US in 2011-2012. We have approximately 105 employees, 35 commercial in North America, 27 commercial in Europe. We feel like we've got the right structure in place to leverage for now. If opportunities arise in the future, change in reimbursements, et cetera, we could increase the investment a little bit, but we feel like we have a pretty good footprint. Direct operations in North America and throughout Europe, partnerships around the globe in places like Australia, Chile, et cetera. We did 393.1 million NOK in 2022 as total revenue. And the product we're going to be talking about today is approved in over 30 countries. And a key part of our strategy going forward is to continue the expansion of blue light technology to become a standard of care worldwide. Next slide, slide 4. So let's just drop back, talk a little bit about bladder cancer. It's really interesting, this space. I joined the company about 5 years ago. Around 5, 6, 7 years ago, bladder cancer in the urology world was not really paid attention to. Yet, I think what surprises most people when you think about cancers is you don't think about bladder cancer being the most expensive cancer to manage for any type of cancer patients out there, highest per patient. The reason for that is because of the 2 bottom parts. The recurrence, 61% up to 1 year, and up to nearly 80% of the folks who have been diagnosed with bladder cancer will have reoccurrence within 5 years. And then progression-wise, in treating cancer, as long as it remains non-muscle invasive, it's treatable through TURBTs, BCG, et cetera, different therapeutics. However, once it becomes muscle invasive, it becomes a very, very different situation. Patients' quality of life and life expectancy plummets. So what's really key in bladder cancer is the ability to detect it and to treat it. And to detect it, you have to see it, and to see it, you have to have the best technology. And that is exactly where Photocure stands with its blue light technology with Cysview and Hexvix. So let's go ahead and talk about that. Next slide, slide 5. So this is, from a business perspective, this is a razor blade model. We, Photocure, sell the item on the left of this slide, which is Cysview or Hexvix. It is a lyophilized powder that's reconstituted. It is instilled into the bladder about an hour prior to a procedure. And then using the equipment on the right, sold by Karl Storz, Richard Wolf, or Olympus, either using a rigid scope if you're in the operating room or a flexible scope if you're in the physician's office, you turn on the blue light, and when the blue light turns on, it visualizes the cancer cells. The cancer cells, to kind of make this simplified, preferentially metabolize our product. And when they do, they glow bright pink under a blue light. This is, this drug device combination brings better visual contrast between malignant and benign tumors. So this technology, as I mentioned, has been out in the markets in the Nordics in 2005, 2006, and then into the US in 2012. Just one other comment about this. In the US, we're relegated to only using or pairing up with Karl Storz equipment. We assist in the selling of the equipment on the right. However, that is products sold by large manufacturers. In the rest of the world, Olympus and Wolf also sell blue light technology. All right, next slide, slide 6. So why is this, why blue light? What does it mean to a patient? Well, I tell you, if I had cancer, bladder cancer, or suspected of bladder cancer, I would surely insist upon using blue light technology with our product Hexvix or Cysview, depending on where you are in the world. Called Cysview in the US, Hexvix, rest of world. The product, as I mentioned, preferentially accumulates in the cancer cells, going bright pink. The difference is up to 21% of recurrent patients are found, 35% in the OR with Hexvix and Cysview. If you think about it, that means about 2 out of 3 patients or 1 out of 4 patients are going to go home believing they're cancer-free under white light cystoscopy. But using blue light cystoscopy with our product, that cancer can be found, the patient can be properly treated and staged, and hopefully have a better outcome, reducing recurrence and hopefully progression. Next slide, please. So let's talk a little bit about the patient journey. It's characterized by a long-term follow-up with repeat procedures. As I mentioned earlier in today's presentation, on the left side, you'll see the blue circle says NMIBC, that's non-muscle invasive bladder cancer. The whole key to treating bladder cancer is to keep that patient on the blue line. When you go to the right side of the page, you'll see MIBC, that's muscle invasive bladder cancer. That's where it's progressing to the stages where it's deep inside the bladder wall and the bladders have to be taken out, or the patient's outcomes, quality of life, et cetera, plummet. The key, though, is treating the patient in the non-muscle invasive stage. So how does a patient get found? Typically, the patient will have some sort of pain, maybe blood in the urine, et cetera. The physician will order up some labs. In addition, while they're in the office, they'll ask the patient, they'll do a cystoscopy. They'll look inside the bladder. This is a local treatment used with white light cystoscopy. They'll look inside the bladder. If they see anything suspicious or the labs come back with some suspicion, they'll book the patient into the operating room. That's where the box, you'll see the box right there in the center, says TURBT, that's transurethral resection of bladder tumor. There are about 700,000 patients between the US and Europe that have these procedures done. It's the ability for the physician goes in, the patient is, as I mentioned earlier, they're put under general anesthetic. In our case, we would want blue light technology to be used with Cysview or Hexvix. The patient would be instilled an hour prior to procedure. The doctor will go in with a cystoscope, turn on the white light, look on the inside of the bladder, then flip to a blue light in which all the cancer cells glow bright pink. The physician then can easily resect the tumor, take out pieces, take biopsies, and at that point they get a proper staging. Are they low grade, high grade, intermediate grade, high risk, low risk? This is extremely critical, this classification, to the patient's ultimate outcome and treatment regime over the course of their life going forward. They move then after they've had the resection, taking out all the tumor that they can see, and of course under using blue light technology with Cysview and Hexvix, they're going to see a lot more of the tumor. They're not going to miss it. And if you miss the tumor, that tumor is going to continue to grow, and of course it can lead to muscle invasive. So it's very important that the physician see all of it, and that's what blue light technology brings to the physician. They then generally will use some sort of chemotherapy, mitomycin, gemcitabine, or BCG, and then they grade and stage the tumor. On the bottom of that, you see the little wheel at the bottom. It says regular surveillance cystoscopies according to risk classification. If you're high risk, you'll be back in 3 months, intermediate risk every 6 months, low risk maybe once a year. If you show remission after 5 years, the physician may say, you know what, I don't need to see you so often. But typically, there are about 1.65 million of these surveillance cystoscopies. Again, if this patient has been resected in the operating room, determined to be high or intermediate grade, coming back for the surveillance in 3 months or 6 months, it is imperative they use blue light technology. Again, if they miss it, then that patient could end up on the right side of this slide, and the prognosis for that patient's recovery is very poor over time. So That's, in total, there are about 2 million of these procedures, 2.3 million of these procedures between Europe and US. Let's go to the next slide. So the key and where we're heading to is developing a new standard of care in bladder cancer. And it takes time, takes a lot of elements, a lot of parts to get it there. I will say that the awareness of blue light cystoscopy with Cysview and Hexvix continues to grow. It is recommended in global and national guidelines. EAU just updated their guidelines and for the first time have specifically listed blue light technology, strengthened it, and talked about the importance of using it. So, and this is an ongoing process for us. It's in the guidelines, but you continue to provide more and more data to strengthen our position. So it is recognized. I think there's been challenges with blue light technology because it is a drug-device combination, and we are reliant upon the device manufacturers to deliver the equipment. And we'll get into that in a moment. Next slide, please. So talking about installations, this is an example in the US of the installations that have been taking place. You see a huge jump up in 2018, 19, 2020, we had COVID. And then in 2021, Karl Storz, again, the US, we're only indicated with the Karl Storz equipment, elected to do a model change, upgrade to their system, which they had sorely needed, and finally got that upgrade out in 2022 into the fourth quarter. So those 59 installations of the new Saphira equipment all happened in the late fourth quarter. And it's important. This is a drug device combination, a razor blade model. It's important to get the razors out there, which is these blue light scopes, because then we can filter in the Cysview or Hexvix, which is the razor blade. Overall, we've got about 380 towers out there. Saphira now represents about 25% of that base. So 25% of the blue light scopes in the US have now been either upgraded or installed as the new blue light technology. And this is a big jump. When the oldest system, going back prior to 2022, or from 2022 third quarter backwards, was an old standard definition. It'd be like looking at a TV from 1985. However, as good as blue light is with Cysview and Hexvix, it did its job. But the visualization was grainy, not quite as good as when you're used to watching LED. That has been upgraded for Karl Storz. What's equally interesting, I think very notable, is when we took back Europe from Ipsen Pharmaceuticals in 2020, the other manufacturers, Olympus and Richard Wolf and Karl Storz, all had sort of lost interest in blue light technology because Ipsen Pharmaceuticals, who sold the product back then, really wasn't selling it. And there had been sort of a loss of interest in it. That has all changed. Richard Wolf has state-of-the-art equipment. Karl Storz has further upgraded their European system, and Olympus will launch a new blue light module system at the end of this year throughout Europe. So the interest has grown. The manufacturers have reinvested in blue light technology in Europe, and I think that's an exciting development. So let's move to slide 10, speaking of Europe. So as I mentioned, when Photocure got the Hexvix approved in Europe, we first started with a partner, GE Medical, and this is in, as I mentioned, 2008 timeframe, 2006 timeframe. GE Medical did a great job, device-oriented company. They decided that they did not want to move forward around the 2010 timeframe, 2011, forward in the urology space. They asked us to find another partner. We found Ipsen Pharmaceuticals. And you notice I emphasize pharmaceuticals. And one thing that you pick up with this product is this is a drug-device combination. It orients itself closer to a device sale. It's a long process. Sale process can go out to a year to get an account up and running on blue light technology. It requires a tremendous amount of experience to navigate the hospital system, the operating rooms. This is a procedure where patients, where our representatives will gown up, go straight through a door that says authorized personnel only, and sit side by side with physician, help guide them through a surgery. So, GE Medical then hands off to Ipsen Pharmaceuticals. Ipsen thought they wanted to be in the urology space. They were developing some products. The problem was that this product did not fit a pharmaceutical sale, it's a device sale. And so they let it basically go backwards. It Sales over time were all going in the negative direction. We took the business back in late 2020 in the midst of COVID. So there has been a lot of work over the last 2 years during the COVID pandemic. It's been challenging, limited access in the first year or two. We've had challenges. We have had, what's striking happening now in parts of Europe, particularly in the UK. There's been a lot of challenges, but what has happened over the last 6 to 12 months is this growing awareness. The KOLs have all been ignited, re-engaged. At EAU, the European Urology Conference, we had the largest, most interest in blue light technology and Cysview and Hexvix in history. It's been amazing to watch the development. So, and again, I mentioned that the equipment manufacturers have all reinvested in blue light technology for Europe. So the interest has actually been reignited. So we look at the business kind of basically in 3 buckets. We look at the established business, the Nordics, with penetration rates of 26 to 58%. We use penetration because there is no direct competition per se of blue light technology. There is not an alternative to white light. To blue light other than white light. And white light, we've already talked about earlier today, is inferior by a large degree. These countries, getting up to 26, 27, 28, 50, 40% is really a high penetration rate. In our mind, once you get up in the 35, 40% range, you've got a really good established business. Then we look at the priority growth markets, France, UK, Italy, Benelux, et cetera, penetration 2 to 14%. These were markets where Ipsen had let erode, and we have now turned them around. We're starting to see growth, stabilization, and reactivation in those markets. And then there's a large untapped section, Spain in particular. We're working with one of the manufacturers to bring in the technology so that we can bring our product into Spain. The reimbursement's in place, Poland, Baltics, et cetera. There's a lot of other opportunities. Now, whether we go direct or use a distributor, we'll assess each individually. But in these countries, there's relatively no penetration, but there's a large opportunity for us. So Europe is really interesting. It's reactivated and looking for growth as we move through the second half of this year and into 2024. Next slide. Slide 11, real quick. Overall TAM, total addressable market, around $2 billion. This is just relative penetration rates of Nordics, DACH, US, and rest of Europe. We believe we have the key success factors in place to become a standard of care. We've reignited the market. The approvals are there, the acceptance on guidelines, permanent and favorable reimbursement. We'll continue to always work on that. Activate awareness. Patients are now aware of it. If you Google bladder cancer, you will come across blue light technology, and patients are actively seeking it. And we've put the right commercial structure behind it. Slide 12, please. So overall, mission, delivering transformative solutions, improve the lives of bladder cancer patients. This is sort of our 4-step strategy. We've been heavily focused on steps 1 and 2, which is single product company, accelerating the growth, expanding sales throughout geographies around the world, partnerships or directly. We're looking now forward to the next phase. We still remain a single product company. It's important to diversify and build out. We have a great commercial infrastructure that can be leveraged with other products or opportunities. So we'll look for the right opportunities. We're not looking to just add anything. It has to be synergistic with blue light technology that'll help strengthen the portfolio and, of course, transform, et cetera, the organization. So I'll finish off with slide 13. So 2023 anticipated milestones, objectives. We indicated 65 to 75 Saphira installs. I will remind you that I didn't get into this flexible cystoscopy versus rigid because this is a very short presentation. But in the world of cystoscopy, the rigid scopes, and then there's the flexible scopes that are used in physician's office. In U.S., Karl Storz has indicated that they are going to no longer provide flexible cystoscopy until a reclassification with the FDA, which will allow them to upgrade that equipment very quickly. So we basically are still saying that we can stay 65 to 75 rigid Saphira installs this year. Product revenue growth above 20% and generate positive EBITDA in 2023. We're going to continue. I mentioned briefly about blue light technology in the US. We're only indicated with Karl Storz. It's a PMA or Class 3 approval, meaning they are only indicated with our product. We're only indicated with their product. Karl Storz has initiated a citizens petition to reclassify the blue light technology system. And if they get and are successful, that will open up the doors for economic and technological competition in the US, which would be a huge change in the market, because right now it is a monopoly for Karl Storz. They've had up until this new Sapphira launch inferior equipment and at a monopoly or premium price. So this will open up the opportunity for Richard Wolf, Olympus, and we actually are aware of about 7 or 8 other companies that want to come to market. So this would be a tremendous opportunity for us if they're successful. That is in process. The 210-day citizens petition was up on May 25th. So we are hopefully going to hear from Karl Storz will hear from the FDA in the next week or so, or not in the next week, over the course of the next months. This process is undefined. It's not statutorily required timeline. Basically, what it says is, at 210 days, the FDA is then has the ability to respond to this reclassification petition by Karl Storz. These typically can take anywhere from under a year to years. Most of them are under 2 years to get reclassified. We believe that there's enough interest and enough reason to believe that we could hear something by the end of this year. So fingers crossed on that. A lot of new data coming out, and of course, we'll continue to leverage opportunities and organizational strengths. So with that, I'll go to Q&A. Last slide.
Thank you, Dan. Great presentation. And first off, I mean, you talked just now quite extensively about this on the sort of reclassification progress, but I just wanted to throw a short question to you on that as well, because typically around 180 days, the comment window is open, and as you said, that was sort of passed now on May 25th. But FDA says that it can be open as necessary, basically, but I haven't seen any sort of specification of what's necessary. I mean, last time I checked, I think there were 18 comments. Should we look at sort of the amount of comments as sort of the necessity aspect of it all, or how do you define when the comments, when there's enough comments, basically?
Yeah, the comments, it's really the quality of the comments and the diversity of those comments. If you look through the comment section, we have it from manufacturers, we have it from providers, we have it from other manufacturers, we have it from patient advocacy groups. I mean, that's what the FDA is looking at. It's not the number, but it's the robustness and the sort of the surrounding stakeholders that write into it. So I feel like we're in a really good place. I think what's interesting is Karl Storz basically, withdrawing Flex out of the U.S. market has put an emphasis on the importance of this reclassification, because Karl Storz, the reason for them doing is, one, the technology is lagging and it's not very reliable. Second part is they cannot upgrade it because of this Class 3. It is extremely expensive and time consuming, takes a lot of resources. If it goes to a Class 2, it's a lot easier through a 510K pathway. So their strategy, if I'm to read through the tea leaves, is that by withdrawing Flex, this will also put pressure on the FDA to make the change so that they can bring this back to the market. Okay, perfect. Yeah, and my next question was actually on Flex as well. I mean, we talked a lot about BLC being used in more of the ambulatory setting as well, and we're seeing a sort of a trend in the US with patients moving there as well. And you also got improved reimbursements in that area also. So Flex was supposed to be quite important as a growth driver going forward. But I was just wondering of basically what you see in terms of timeline after the sort of reclassification has been made. Do you see it as feasible to coming back with Flex in a sort of relatively short time period? Yeah. So a couple things. One is, we're working hard behind the scenes to get control of our own destiny with surveillance or flexible cystoscopy, and that's in the form of finding someone who will manufacture called a proprietary system for photocure, and we'll work on that. In the meantime, yes, if that reclassification comes through, a couple things will happen. One is we're fully aware on the rigid side that people like Wolf, Olympus, and others are prepared to submit for an approval in the U.S. On the flexible side, I think, this is where I think we need to have control of our own destiny, prove out the technology, prove the importance. I will tell you this, on the surveillance side with flexible cystoscopy, those accounts that had flex, and it was growing for us, particularly, you know, if you think about, we launched in 2019 and then hit COVID, but still it was a growing interest. The data behind it continues to grow. Those accounts were far more productive in terms of how quickly they could get to 200, 300, 400 units versus a rigid account, you know, a hospital-based account, because they see so many more patients. They see 3 to 4 times more patients, so greater opportunities. And there's more pressure from a you know, reimbursement side and treatment side of putting more and more procedures in the physician's office. So we think we're going to come back together at a good time where flex, hopefully we, you know, with a reclassification, we could get something out in the next, you know, 2 years or under.
Okay, perfect. And then I've noticed, especially now in Q1, that your sort of market comments on CapEx demand from your customers, you mentioned, you talked about a little bit of a softer environment than maybe your peers did in other areas. So I was just wondering if you think that your customers maybe see blue light as, let's call it more of a luxury, more nice to have rather than need to have, than maybe what's seen in other areas? Or do you see something specifically for urology that's a weaker area overall?
Yeah, well, we get hyper specific on market trends. So we're looking at TURBTs. We buy claims data. So when you hear things like, oh, the urology market is either good, up or down, or otherwise, you know, if you got to get below that surface to see specifically what's going on. What we're seeing in the TURBT market is that there has been a softening of the market over the last couple of years. I think some of that's attributed, to be honest with you, is, you know, especially in the surveillance side, it's not a must-have procedure like that moment. It's not a critical, you know, procedure to have. They can postpone it by a month or 2 or 3. So I think that's part of what's happened here. Blue light cystoscopy, or the, you know, is it a luxury or not? I think that, in my opinion, I don't believe that physicians are treating that way. What they are doing, though, is saying, where is it, you know, where's it important to get it in? If you're going to do that one cystoscopy, and we try to make this point with them, if you're only going to get one cystoscopy in, make it a blue light cystoscopy. Make it, you know, the best technology so you get the right diagnosis and treatment afterwards.
Yeah. And then I think the last question that we have time for, I mean, we've seen over the past year or two now sort of a negative trend in utilization per scope. Even now with like overall procedures trending fairly positively, at least on the data I see. So I was just wondering if you see any specific reasons for the lower utilization. Is that primarily, you know, new scope placement that takes some time to ramp up to speed, or is there anything else more broad going on?
Yeah, it's actually 2 parts of the equation. One is that, especially last year, if you try to do the analysis, most of the equipment installs happened in the 4th quarter, you know, 40-something. So you take them and you put them in the denominator, it throws it way off. They didn't have enough time to produce anything. In fact, they didn't really produce anything until starting Q1 and going forward. The second part of this is just the dilemma we face, is particularly in the U.S. with the, you know, this class 3 level of approval. The you know, Karl Storz's equipment in the U.S. and the European equipment, you know, more recently as well, is old and it's not quite reliable. So there are accounts that have had challenges, but not all accounts. There's accounts that have grown. There's definitely accounts that have had challenges with their equipment. They want upgrades. They want more reliable equipment. So, but, you know, I think it's a 2-part question, but I think, you know, one is the denominator, the math behind it. And I think you got to keep that in mind. But the other one is, I think, just the equipment, I think, will solve some of that challenge we've had in the background.
Okay, perfect. I just came up with a question, so I want to squeeze that last one in. Now that Sapphire has been on market for a little while, have you seen any, you know, sort of differences in utilization between the old generation and the new one?
We have a lot of what do you call it? What the word is, but you know, off the cuff comments. We know some examples, like in France, there was an account that put the new equipment in, had 70% improvement in utilization. We thought that there's going to be a couple things. One is better visualization, more excitement. Physicians want to use it. It's easier to use. It's more reliable. Cleaning it's a lot easier. You're not going to have as many breakdowns and things like that. We think that's going to add a lot to the utilization. What we're going to do is we are measuring in the background, but you need some longitude longitudinal data to this. If I pick 1 month over another month, it could be a false positive or a false negative. But what we are seeing is a general trend to increase utilization. So we look forward to reporting that out later on this year and see how this develops.
Perfect, perfect. Well, thank you very much, Dan. This is the last presentation for today, so also thank you to everyone listening in as well, and have a good rest of the day.
Great, thank you.